Right now, in parts of the ocean deeper than most submarines can safely reach, something is moving through water the sun has never touched, and it has been down there for 400 million years. Long before the first tree existed on land, before the dinosaurs, before complex life had fully settled onto the continents, the ancestors of this animal were already moving through the deep ocean. Most of the creatures that shared its world are gone now. This one stayed. It is called a chimaera, a relative of sharks so ancient that it split away from them before sharks had fully become what we recognize today. Its body looks assembled from mismatched parts: large reflective eyes, wing-like fins, a tail that narrows into something almost whip-like, a venomous spine rising from its back, and a skeleton made entirely from cartilage, with not a single true bone anywhere inside it. Some species have never been filmed alive. Most of what scientists know comes from damaged specimens pulled accidentally from deep fishing nets, brought to the surface from depths where pressure, darkness, and cold have remained almost completely unchanged for hundreds of millions of years. And hidden inside the genome of this animal are genetic sequences that disappeared from nearly every other vertebrate lineage on Earth, including ours. The deeper researchers study the chimera, the stranger it becomes, not because it behaves like a monster, but because it seems to belong to an earlier version of the planet itself, a world that no longer exists anywhere except in the deepest water on Earth. By the end of this video, you will understand why the chimera may be one of the most important animals in the ocean, how it survived nearly every extinction in Earth's history, and why scientists believe it may still carry clues to the original blueprint of the vertebrate body itself. Long before scientists agreed on what to call it, the animal already seemed assembled from contradictions. Its body carries the outline of a shark, yet the face narrows into something almost mammalian, soft and uncertain in the darkness. The fins open outward like folded wings drifting beneath the water, while the tail extends behind it in a thin tapering line that appears less like a fish and more like a strand of living wire disappearing into blackness. Even in preserved museum specimens, drained of movement and color, the chimera retains the impression of something unfinished by ordinary evolutionary standards, as though several ancient creatures had quietly dissolved together in the deep ocean and emerged as a single form. Scientists place chimeras within the class Chondrichthyes, the ancient group that also contains sharks and rays. Beneath that broader classification, however, the separation becomes immense. Chimeras belong to their own subclass, holocephali, a lineage that diverged from the ancestors of modern sharks roughly 400 million years ago during the Devonian period, when vertebrate life was still experimenting with body plans that no longer exist on Earth. In practical terms, this means a modern shark is not the chimera's close relative in the way most people imagine. Both lineages simply inherited the same distant cartilaginous foundation before evolving along radically different paths for hundreds of millions of years beneath changing oceans and collapsing worlds. Today, approximately 50 living species are formally recognized, though the true number may be higher in the unsurveyed regions of the deep sea. These species are divided into three surviving families. The chimaeridae are often called ratfish because of their tapering tails and rounded snouts. The rhinochimaeridae, the long-nosed chimeras, possess elongated faces that project outward like delicate spears. The calorhentidae, sometimes known as plow-nosed chimeras, carry broad fleshy snouts shaped almost like submerged tools designed to sift through sediment. Across all three families, the body remains unmistakably ancient. Most species measure somewhere between 60 centimeters and 1.5 meters in length, Though certain deep water forms approach 2 meters from head to tail, their appearance becomes stranger the longer it is observed. The skin is smooth and scaleless, pale in some species and charcoal dark in others, often carrying a faint iridescent sheen beneath artificial light. The pectoral fins dominate the body, broad and flexible, moving with a slow airborne rhythm, unlike the muscular undulation of most sharks. The eyes are unusually large, reflective green beneath illumination, adapted for conditions where almost no sunlight survives. Some snouts remain blunt and rounded, while others stretch forward into narrow projections that seem almost detached from the rest of the skull. The name chimaera emerged from Greek mythology, borrowed from the chimera, the hybrid creature composed from incompatible animal parts. Early naturalists saw the resemblance immediately. Most chimeras inhabit depths between 200 and 2,600 meters below the surface, though scattered records suggest some descend even farther into darkness that remains almost entirely unvisited by human observation. At those depths, sunlight disappears completely, pressure rises to immense levels, and the ocean settles into near-freezing stillness. Because of this isolation, nearly everything known about chimeras has historically come from dead individuals accidentally dragged upward in trawl nets. Many arrived damaged from decompression, pale and motionless on steel decks, before scientists ever saw them intact. For most of human history, the chimera existed less as a living animal than as fragments of anatomy carried briefly out of the deep, offering small glimpses of a world that remained hidden beneath the surface. The ocean of the Devonian did not resemble the modern world in any familiar way. Continents existed as scattered masses still drifting toward future collisions, and across the southern half of the planet stretched the immense supercontinent Gondwana, a landscape barren of forests and quiet beneath skies only beginning to fill with oxygen in concentrations approaching those of the present Earth. Rivers carried sediment into shallow inland seas that spread across much of the globe, warm and mineral-rich beneath a younger sun. Along the coasts, the first primitive plants were beginning to anchor themselves to wet ground, thin green growths no taller than a person's knee, while offshore the water had already become the center of a vast evolutionary acceleration unlike anything the planet had previously known. For this reason, the Devonian is often called the Age of Fishes. Life in the sea was diversifying with extraordinary speed, experimenting with jaws, fins, armor, and sensory systems in forms both elegant and brutal. Coral reefs stretched for enormous distances beneath the surface, crowded with trilobites, ammonoids, and early vertebrates moving through suspended clouds of plankton. Some fish remained small and delicate, drifting through open water in silver schools, but others became massive armored predators called placoderms, creatures wrapped in thick bony plates that turned their skulls and torsos into living fortresses. Among them swam Dunkleosteus, one of the great hunters of the Devonian Sea, reaching lengths of nearly 9 meters. Instead of teeth, its jaws carried sharpened bone plates capable of shearing through flesh and armor alike, with a bite force estimated at roughly 8,000 pounds per square inch, strong enough to split the bodies of other large fish in half with a single closure of the jaw. It was within this crowded and violent ocean that the ancestors of the chimera began separating from the lineage that would eventually produce modern sharks. At first, the distinction may not have appeared dramatic. Both groups possessed skeletons of cartilage rather than bone. Both relied on ancient sensory systems suited to underwater life, and both emerged from the same broad radiation of early jawed vertebrates. Yet over immense stretches of time, their trajectories quietly diverged. The ancestors of sharks moved towards speed, muscular propulsion, and pursuit predation in open water. The ancestors of chimeras drifted in another direction entirely, away from the brighter surface ecosystems and toward deeper habitats where movement slowed and food became sparse but steady. Their bodies adapted for crushing shelled invertebrates along the seafloor rather than chasing fast prey through illuminated water. The decision was not conscious, only the gradual shaping force of survival acting across millions of years, but the consequences became enormous. Every placoderm that dominated the Devonian Ocean disappeared. Dunkleosteus vanished. Entire reef systems collapsed. The armored giants that once ruled the seas survive now only as fragments of fossilized plate buried inside stone. Yet the chimera lineage persisted quietly beneath extinction after extinction, carrying its ancient anatomy forward, while dominant groups around it rose briefly and disappeared. The first great collapse arrived during the late Devonian extinction, when changing climates and declining oxygen levels destabilized marine ecosystems across the world. Much later came the Permian-Triassic extinction, the most severe biological catastrophe Earth has ever experienced, eliminating approximately 96% of marine species. Then followed the Triassic-Jurassic extinction, the asteroid impact at the end of the Cretaceous that erased the non-avian dinosaurs, and eventually the modern extinction unfolding across the present oceans. Through all of it, the chimera endured. The final Tyrannosaurus rex disappeared 66 million years ago, a span of time so immense that human history becomes almost invisible beside it. Yet the lineage of the chimera stretches back more than 6 times farther, reaching into seas that existed before forests, before birds, before flowering plants, before the shapes of the continents themselves had settled into the arrangement now visible on a modern globe. Inside the body of the chimera, there is no bone at all. Beneath the skin, beneath the drifting movement of the fins and the strange reflective eyes, the entire internal framework is built from cartilage, a softer and more flexible material familiar in the human ear, the tip of the nose, and the joints where bones meet and glide against one another. In most animals, this material exists only in supporting roles, temporary scaffolding, eventually replaced by hardened skeleton during development. In the chimera, it remained. 400 million years after its lineage separated into the deep ocean, the body still carries the architecture of an earlier vertebrate world. This matters because the vertebrate skeleton did not appear all at once. Long before animals evolved dense mineralized bone, capable of supporting enormous land bodies or powering muscular limbs across continents, there existed softer internal structures suited to ancient seas. The chimera preserves part of that older design. Sharks and rays share the same cartilaginous ancestry, yet the chimera diverged so early that its body may resemble conditions closer to the common ancestor of all jawed vertebrates than almost any other living animal now swimming on Earth. In laboratories and museum collections, researchers study the mineral patterns inside chimera cartilage with unusual attention, searching not only for information about the fish itself, but for clues about how skeletons first emerged in evolutionary history. The difficulty is that cartilage rarely survives the passage of geological time. Bone fossilizes comparatively well because its dense mineral structure resists decay long enough to become buried and preserved inside sedimentary rock. Cartilage collapses quickly after death, consumed by bacteria, currents, and scavengers before fossilization can occur. For this reason, the chimera leaves behind only fragments of its existence. Entire oceans passed over this lineage, while the physical evidence vanished almost completely. Most fossils attributed to ancient chimeras consist of isolated tooth plates or dorsal spines scattered through rock layers, separated by millions of years. Full skeletons remain extraordinarily rare, and vast portions of the lineage's evolutionary history survive only as absence. The teeth endure because they are unlike ordinary cartilage. Chimeras do not possess rows of replaceable cutting teeth like sharks. Instead, the jaws contain three pairs of permanent tooth plates, two embedded in the upper jaw and one in the lower. These plates are heavily mineralized and continue growing throughout the animal's life, slowly thickening as they grind against one another. Their surfaces become polished and worn through constant pressure, forming structures more similar to millstones than blades. When the chimera feeds along the seafloor, the jaws close with crushing force, designed for hard-shelled prey, sea urchins, mollusks, crustaceans, and buried invertebrates, protected by rigid exteriors. It is a quieter feeding strategy than the pursuit predation associated with sharks. Slower and more deliberate, shaped for a world where energy must be conserved carefully in cold water and darkness. Some fossil tooth plates recovered from Devonian rock formations date back roughly 380 million years. Their shapes vary across extinct species, some broad and flattened, others curved into unusual spirals. But the underlying pattern remains recognizable across immense stretches of time. The continuity is striking. Long after entire marine ecosystems disappeared, the tooth plates remained, pressed into stone like signatures left behind by a lineage that almost never fossilized completely. As researchers began examining how these structures mineralize inside living chimeras, another layer of significance emerged. The cartilage is not entirely soft. Tiny deposits of mineral accumulate through developmental processes that resemble the earliest stages of bone formation in higher vertebrates. Some evolutionary biologists now argue that the chimera represents the closest living approximation of the ancestral jawed vertebrate condition, a surviving reference point from before skeletons became fully ossified. In this sense, the animal drifting through the deep ocean today still carries within its body a record of how vertebrate anatomy itself first began to harden, strengthen, and transform hundreds of millions of years before the appearance of humanity. Just ahead of the chimera's first dorsal fin, rising from the back like a narrow blade of polished stone, sits a single rigid spine. In many species, it appears almost ornamental at first glance, too slender to dominate the silhouette of the animal, yet beneath the skin, the structure anchors deeply into the musculature and connects to a venom gland at its base. It remains one of the oldest defensive systems carried by any living vertebrate lineage, quiet and largely invisible until something attempts to seize the fish from above. The spine itself is composed of heavily mineralized tissue, harder and denser than the surrounding cartilage that forms the rest of the chimera's body. Along its surface run subtle grooves through which venom can enter a wound during contact. Unlike the fangs of snakes or the stingers of jellyfish, the chimera's spine is not an active weapon in the ordinary sense. The animal cannot thrust it forward or direct it toward prey. It simply exists along the dorsal surface, angled backward, so that pressure from a bite or grasp forces the spine into whatever has attempted to capture the fish. In the darkness of the deep sea, where encounters are infrequent and energy is carefully conserved, this passive form of defense may be enough. The chimera does not hunt with venom. It does not paralyze prey or use toxins to overpower smaller animals. The spine serves only as protection against predation, a final barrier between the fish and the mouth of something larger moving through the cold water above it. Even this simple arrangement appears remarkably ancient. Fossilized dorsal spines, attributed to extinct chimera relatives, have been recovered from rock layers dating back hundreds of millions of years, suggesting the structure emerged early in the lineage's history and endured through repeated cycles of extinction and survival. Most human encounters with the venom occur accidentally aboard fishing vessels. Deep-sea trawl nets occasionally bring chimaeras to the surface, tangled among rough fish, grenadiers, and other species hauled upward from dark continental slopes. Handlers unfamiliar with the spine sometimes grip the animal carelessly while removing it from the net, only to receive a puncture wound through gloves or exposed skin. Reports from fishermen describe immediate intense pain radiating outward from the wound site, often accompanied by swelling, redness, and lingering discomfort lasting several hours. In some cases, nausea, dizziness, or weakness followed the injury, though severe long-term effects appear uncommon. Because live specimens are so rarely available for laboratory study, much of what is known about the venom still comes from scattered medical reports rather than controlled biochemical analysis. The spine begins developing early in life. In certain species, it is already visible at birth, as a small hardened projection emerging from the dorsal region. As the fish matures, the spine thickens and calcifies further, gradually becoming more rigid with age. In older individuals, the structure can appear weathered and worn, marked by scratches and erosion from years spent moving through sediment-rich water and narrow rock spaces along the seafloor. Some preserved specimens carry spines so heavily calcified that they resemble carved ivory more than living tissue. The chemistry of the venom itself remains poorly understood. Only a handful of chimera species have undergone even partial toxin analysis, and many have never been studied at all. The difficulty lies partly in geography and pressure. Most chimeras inhabit depths where capturing live-healthy individuals is technically difficult, and once brought rapidly to the surface, their bodies often deteriorate before detailed physiological work can begin. Entire components of the venom system may therefore remain undocumented, simply because the animals carrying them have never been examined alive. Among cartilaginous fish, venom evolved more than once. Stingrays possess their own defensive spines equipped with toxins, yet anatomical studies suggest the chimera's system developed independently along a separate evolutionary path. Deep within this ancient class of fishes, separated by hundreds of millions of years, different lineages arrived at similar solutions to the same problem, surviving long enough in dangerous water for defense itself to become written permanently into the body. Between the eyes of a male chimera, recessed into a shallow groove along the forehead, rests one of the strangest anatomical structures in the vertebrate world. At first glance, it resembles a small folded appendage pressed flat against the skull, easy to mistake for part of the skin or an extension of the snout. In preserved specimens, it often appears inert and almost artificial, but in life this structure can extend outward from the forehead as a movable organ known as the frontal clasper, or tentaculum. No other living vertebrate possesses anything quite like it. Despite its unusual placement near the head, the frontal clasper has nothing to do with feeding or sensory perception. It is entirely reproductive in purpose, evolved solely for mating in the dark depths where direct observation is almost impossible. Male chimeras use the frontal clasper as a gripping structure during copulation, anchoring themselves against the female while positioned close together along the seafloor. The organ operates alongside two additional pairs of claspers located near the pelvic fins, giving the male a total of 3 separate clasping systems distributed across the body. Among all known vertebrates, this arrangement appears unique to chimeras, another remnant of a lineage that diverged so early from other jawed animals that its reproductive anatomy followed its own isolated path through evolutionary time. The frontal clasper itself is covered with tiny recurved hooks called denticles, hardened structures capable of catching against the female's skin during mating. Under magnification, these denticles resemble miniature teeth, curved backward in careful rows across the surface of the organ. Exactly how the frontal clasper functions mechanically remains uncertain, because mating has almost never been witnessed directly in the wild. Most assumptions come from dissections, preserved anatomy, and rare observations of captive elephant sharks in shallow research environments. Deep-sea species continue their reproductive lives almost entirely beyond human sight, carrying out behaviors that science still reconstructs largely from inference rather than observation. Like sharks and rays, chimaeras reproduce through internal fertilization. After mating, the female produces eggs enclosed within a durable, leathery capsule, often compared to a mermaid's purse. These capsules drift far from the transparent gelatinous eggs associated with many shallow water fish. Instead, they appear dense, elongated, and carefully sealed against the immense pressures and cold conditions of the deep ocean floor. In some species, the capsule reaches lengths of nearly 25 centimeters, large enough to occupy a significant portion of the female's body before deposition. Freshly laid capsules are usually amber brown or deep golden in color, with ridged surfaces running lengthwise from one end to the other. Some taper into thin, filamentous extensions that may help anchor them against sediment or rocky surfaces, preventing currents from carrying them away. Once released onto the seafloor, the eggs receive no further parental care. The female departs, leaving the developing embryo alone within its protective casing, while cold water moves slowly across the outer membrane for months beneath the surface. Inside the capsule, development proceeds with extraordinary slowness. In certain deep water species, the incubation period extends beyond 12 months, among the longest known developmental cycles for any marine fish. The embryo grows gradually in darkness, nourished by its yolk supply, while organs, fins, sensory systems, and cartilage skeleton form inside the enclosed chamber. At the low temperatures common across deep ocean habitats, metabolic processes remain reduced, stretching development across seasons in near stillness. By the time the young chimaera finally emerges, much of its anatomy is already prepared for independent survival. The dorsal spine may already be present in reduced form, the tooth plates beginning to mineralize, the large eyes adapted for dim conditions long before they encounter open water. There is no school to join and no parent waiting nearby. The juvenile descends immediately into the same ancient world, inhabited by its ancestors for hundreds of millions of years, carrying within its body structures so unusual that even now, after centuries of study, portions of their purpose remain only partially understood. In the beam of a submersible light, the eyes appear first, before the outline of the body becomes visible against the dark water. Two circular reflections flare green and blue-green beneath the surface, suspended motionless for an instant, like distant glass lanterns drifting through the deep. The effect comes from a reflective layer behind the retina called the tapetum lucidum, a structure shared with many nocturnal animals on land, including cats whose eyes flash brightly under headlights at night. In the chimaera, this layer transforms the scarce light of the deep ocean into something usable, capturing each fragment twice before allowing it to disappear back into darkness. At depths below 200 meters, sunlight weakens rapidly until only traces remain. Farther down, illumination comes almost entirely from living organisms themselves, pulses of bioluminescent blue released by drifting plankton, jellyfish, crustaceans, and fish moving invisibly through the water column. The tapetum lucidum reflects incoming light back through the retina a second time, effectively doubling the chance that photoreceptor cells will detect it. In evolutionary terms, even the faintest increase in visual efficiency becomes valuable in an environment where a single flash may reveal food, terrain, or movement nearby. The eyes of the chimaera are enormous relative to the size of the animal. Among deep-sea fish, they rank among the highest eye-to-body proportions known, occupying a substantial portion of the skull and giving the face its unmistakable appearance. Large pupils allow more light to enter, while the broad retina maximizes sensitivity under dim conditions. In preserved specimens, the eyes often appear cloudy and collapsed from decompression, but in life they possess an iridescent clarity that underwater footage struggles to fully capture. The glow is not self-generated. It is the result of borrowed light, returned outward from within the eye itself. Unlike many vertebrates, the eyes cannot rotate independently inside the skull. They remain fixed in place, embedded firmly within the head, which means the chimaera must redirect its entire body or tilt its head to change its field of vision. The movement is subtle and deliberate, a slow adjustment rather than the rapid darting gaze associated with surface predators. In the stillness of deep water, where distances collapse into darkness only a few meters away, sudden visual tracking matters less than broad awareness of the surrounding environment. Vision, however, is only one part of the sensory world the chimaera inhabits. Running across the head and snout lies a network of fluid-filled canals known as the lateral line system. These canals detect pressure changes and vibrations passing through the water, allowing the animal to sense movement without relying on sight at all. A buried crustacean shifting beneath sediment, the pulse of another fish swimming nearby, the subtle turbulence left behind by motion through cold water, all of these disturbances create information the lateral line can interpret. In darkness, where the eyes become limited by distance and available light, vibration itself becomes another form of perception. Sharks possess specialized electroreceptors called ampullae of Lorenzini, capable of detecting weak electrical fields generated by muscle contractions and heartbeats. Chimaeras do not appear to possess this exact system. Instead, they carry an analogous structure known as the rostral organ distributed across the snout. Small pores connect to internal sensory canals, whose full function remains only partially understood. Anatomical evidence suggests the organ may detect low-frequency vibrations and possibly faint electrical signals produced by prey hidden beneath sediment, though the sensitivity of the system has never been fully characterized in living deep-water species. Together, these sensory adaptations form a body designed not for speed or aggression, but for navigation through a world where almost nothing can be seen directly. Giant reflective eyes gather fragments of bioluminescent light. The lateral line interprets invisible vibrations moving through water, and the rostral organ searches the sea floor for concealed life beneath layers of sediment. In complete darkness, the chimaera continues moving through the deep with a sensory architecture refined across hundreds of millions of years beneath oceans that rarely reveal themselves to the surface world. When a shark moves through water, the entire body participates. Muscles contract in waves from head to tail, pushing the animal forward through powerful lateral undulations that ripple visibly along the spine. The chimaera travels differently. Watching one drift across the seafloor feels less like observing a fish swim and more like watching a bird suspended underwater, wings opening and folding through cold darkness with measured precision. The body itself remains comparatively still while the enormous pectoral fins provide almost all forward motion, rising and falling in a slow, continuous rhythm that carries the animal through the deep with very little visible effort. This form of movement is known as rajiform locomotion, a style shared with rays and some skates, though the chimera performs it with a distinctive softness that reflects the conditions of its environment. In the deep ocean, food rarely appears in abundance. Energy lost unnecessarily may not be replaced quickly. Evolution in such habitats favors conservation over speed, steadiness over explosive motion. The chimera's locomotion reflects this perfectly. Rather than accelerating through open water in pursuit of prey, it glides gradually above the sediment, maintaining momentum with minimal muscular expenditure while searching the seafloor below. The long tapering tail, trailing behind the body, contributes surprisingly little to propulsion. In sharks, the tail acts as the primary engine, generating thrust through broad, sweeping movements. In the chimera, it functions more like a rudder, stabilizing direction and making subtle adjustments, while the pectoral fins perform the real work of movement. The tail often drifts almost motionless behind the body, following the animal through the water in gentle curves, as though carried by inertia rather than force. Underwater footage captured by remotely operated vehicles reveals how little disturbance the chimera creates as it moves. Sediment barely rises beneath it. The surrounding water remains calm, almost undisturbed, except for the slow oscillation of the fins. There is no violent acceleration, no muscular thrashing associated with pelagic predators crossing open oceans near the surface. Even when changing direction, the movement remains fluid and economical. The fish seems less to swim through the water than to settle into it, adjusting its position gradually as though perfectly matched to the density and pressure of the environment around it. Part of the unusual atmosphere surrounding chimera footage comes from this restraint. Many deep-sea organisms move abruptly or unpredictably when illuminated by artificial lights, reacting to unfamiliar intrusion from above. Chimaeras often continue gliding at the same unhurried pace, crossing the field of view with a composure that feels almost detached from urgency. Their movement carries the impression of an animal that has existed for so long in stable darkness that speed itself became unnecessary. Across hundreds of millions of years, there was no evolutionary demand to hurry. Most species also appear to occupy relatively localized ranges rather than undertaking the vast migrations associated with many shark species. Great white sharks, whale sharks, and hammerheads may cross entire ocean basins over the course of a year, navigating thousands of kilometers between feeding and breeding grounds. Chimaeras seem more geographically restrained, remaining within specific continental slopes, submarine canyons, or deep shelf regions for extended periods. Precise tracking remains difficult because attaching electronic tags to fragile deep-sea fish presents enormous technical challenges. Yet, available evidence suggests many species spend much of their lives within limited territories along the seafloor. Some species, however, appear to perform slow vertical migrations linked to darkness above. During nighttime hours, they may ascend into shallower water to feed, taking advantage of prey organisms that rise through the water column after sunset. With daylight, they descend once more into deeper zones where temperature and light remain stable. This behavior has only been documented in a small number of species, but it hints at a hidden rhythm unfolding continuously beneath the ocean surface. A gradual movement, upward and downward, tied not to tides or seasons, but to the faint shifting boundary between darkness and the last traces of light filtering down from the distant world above. Across the deep sea floor, food rarely appears in sudden abundance. It exists instead as scattered pockets of life hidden beneath sediment, tucked between rocks, or drifting slowly across the bottom in the darkness. The chimaera moves through this landscape with patient attention, searching not for large prey, but for durable organisms that spend their own lives close to the ocean floor. Sea urchins, crabs, mollusks, polychaete worms, and occasional small fish make up much of its diet, all gathered gradually from the benthic world beneath it. The feeding process begins long before the prey becomes visible. As the chimaera glides low over the seafloor, the sensory systems distributed across its head interpret disturbances hidden within the sediment below. The lateral line detects minute vibrations traveling through water and substrate, while the rostral organ along the snout appears capable of identifying faint biological signals produced by buried organisms. In this way, the fish searches the ocean floor almost by touch at a distance, reading movement through pressure and subtle electrical changes in the environment around it. When prey is located, the chimaera lowers its head toward the sediment and begins probing carefully along the bottom. Some species use the flexible snout to disturb loose substrate, exposing buried invertebrates beneath layers of fine marine debris accumulated over centuries. The motion remains slow and deliberate. There is little wasted movement, no sudden burst of acceleration. Once uncovered, the prey is drawn inward and positioned between the mineralized tooth plates that line the jaws. Those plates generate considerable crushing force relative to the size of the animal. Sea urchins protected by rigid calcium shells, crabs enclosed within hard exoskeletons, and thick-shelled mollusks all require substantial pressure to break apart. The chimaera accomplishes this not through sharp cutting teeth, but through compression and grinding. The upper and lower tooth plates meet together like the surfaces of a millstone, slowly pulverizing shells into fragments while softer tissue is consumed. The process differs entirely from the feeding mechanics of predatory sharks, whose serrated teeth slice flesh through repeated biting motions. The chimaera feeds through pressure rather than tearing, transforming armored prey into manageable pieces through patient force. Stomach analyses from specimens brought up in deep-sea trawl nets have revealed surprising variation in diet between individuals. Some chimeras contained large quantities of echinoderms, such as sea urchins and brittle stars, while others appeared to feed primarily on crustaceans. Certain specimens carried remains of worms and small fish in greater proportion than expected. This variation may reflect differences in local prey availability across deep ocean habitats, or it may suggest individual feeding preferences shaped by geography, depth, and competition along particular sections of the seafloor. Food scarcity at depth shaped another defining characteristic of the lineage, an extremely slow metabolism. In cold environments where prey may be widely dispersed and encounters infrequent, rapid energy consumption becomes dangerous. The chimaera survives partly because it requires comparatively little. Long intervals between successful feeding events appear tolerable, allowing the animal to persist in ecosystems where productivity remains low and unpredictable. Across geological time, this efficiency may have become one of the lineage's greatest advantages. During periods when marine ecosystems collapsed and faster metabolisms failed, a body adapted for minimal expenditure could continue functioning quietly in the background of extinction. Yet, despite centuries of study, much about the chimaera's ecological role remains unresolved. Almost nothing is known about how different species compete with one another for food in the deep ocean. It is unclear whether they actively avoid overlap with deep-sea sharks, rays, or bony fish occupying similar habitats, or whether the vastness of the environment reduces direct competition altogether. Even their predators remain uncertain. Larger sharks may occasionally prey upon them, and marine mammals capable of diving to great depths could potentially encounter them. But direct observations are extraordinarily rare. Most of the chimaera's feeding life therefore unfolds unobserved, carried out slowly across dark seafloor plains far beyond ordinary human reach, where movement through sediment and the soundless crushing of shells continue much as they have for hundreds of millions of years. Far beneath the shipping lanes, coastlines, and storms of the surface world, the chimaera inhabits nearly every major ocean basin on Earth. Cold, deep water connects regions that appear separated on maps, and across those submerged expanses, chimeras persist in the Atlantic, Pacific, Indian, Arctic, and Southern Oceans. Few groups of deep-sea fish possess such a broad, global distribution. The animals themselves remain rarely seen, yet their lineage extends quietly around the planet beneath continental shelves, submarine slopes, and abyssal plains where sunlight disappears and human observation becomes difficult. Despite this global reach, individual species tend to remain confined to specific ocean regions with remarkably little overlap. One basin may contain species found nowhere else on Earth, separated from distant relatives by immense stretches of deep water that function less as open highways and more as evolutionary barriers. Over geological time, populations isolated along separate continental margins gradually diverged into distinct forms. In the deep ocean, distance itself becomes a force shaping anatomy, genetics, and behavior across millions of years. Along the Pacific coast of North America lives the spotted ratfish, Hydrolagus collier, among the best known of all chimeras because it occasionally appears in shallower waters accessible to scientific observation. Its range extends from Alaska southward toward Baja California, where it inhabits cold continental shelf environments and deeper offshore slopes. In underwater footage, the species appears pale silver with scattered darker markings across the body, moving close to the seafloor beneath coastal Pacific currents that have remained cold and nutrient-rich for countless generations. Far away in the northeastern Atlantic and portions of the Mediterranean swims Chimaera monstrosa, commonly called the rabbitfish. The species inhabits depths along European continental margins, where trawl fisheries occasionally recover specimens from dark seabed habitats. Its broad head and tapering body contributed to the early impressions that inspired many of the chimera's unusual common names. Sailors and fishermen encountering these animals unexpectedly often described them less as fish and more as fragments of unfamiliar anatomy assembled together from different creatures. In the southern hemisphere, another branch of the lineage persists in coastal waters surrounding southern Australia and New Zealand. Cartilaginous millii, often called the elephant shark or plow-nosed chimera, differs from many deep-sea relatives by inhabiting comparatively shallow environments. Its flexible, fleshy snout projects forward like a soft, curved appendage used to probe sediment for buried prey. Unlike most chimeras confined to depths difficult for humans to access, Cartilaginous millii sometimes enters coastal regions shallow enough for direct capture and long-term observation. This accessibility transformed the elephant shark into the most scientifically important chimera species known. Nearly everything understood about chimera physiology, reproduction, cartilage development, immune systems, and genetics comes primarily from this single animal. Most deep-sea species deteriorate rapidly when brought to the surface, their bodies unable to tolerate decompression and changing temperature. Cartilaginous millii survives captivity far more successfully than its relatives, allowing researchers to study living tissue, embryonic development, and long-term biological processes impossible to observe in deeper species. In many laboratories around the world, the elephant shark became the representative through which an entire ancient lineage is interpreted. Even now, however, the catalog of known chimaeras remains incomplete. Since the beginning of the present century, at least 5 additional species have been formally described from surveys in the Pacific and Indian Oceans, many discovered only after preserved specimens from deep trawl operations reached museums and research collections. Careful examination of snout proportions, tooth plate structure, fin placement, and genetic sequences revealed animals distinct from any previously documented species. The process continues slowly because vast portions of the deep ocean remain poorly surveyed. The deepest confirmed sighting recorded so far captured a chimaera at approximately 2,600 meters beneath the eastern Pacific. At such depths, the surrounding environment approaches perpetual night under immense pressure and near freezing temperature. Yet anecdotal reports from commercial trawl crews operating even deeper suggest additional species may inhabit zones beyond regular scientific observation. Their bodies continue moving through water almost untouched by direct study, spread across the hidden geography of the global ocean, where darkness itself still limits the boundaries of human knowledge. In laboratories far removed from the deep ocean, the body of the elephant shark eventually became something else entirely, a map of ancient inheritance written not in cartilage or teeth, but in sequences of molecular code. In the year 2014, researchers completed the first full genome sequence ever assembled from a cartilaginous fish, using Cartilaginous milii as their subject. For chimaera research, the achievement marked a turning point. Until then, much of the lineage's history had been reconstructed through scattered fossils, anatomy, and indirect evolutionary comparisons. The genome offered something far more intimate, the internal record preserved inside every cell of the animal itself. A genome sequence is, in essence, a complete catalog of an organism's DNA, the encoded biological instructions responsible for constructing tissues, regulating development, shaping immune responses, and maintaining life across generations. By comparing these sequences between species, scientists can identify which genes have been retained over evolutionary time, which have disappeared, and which emerged later in entirely new forms. Hidden within those comparisons is a history extending far deeper than written records or even fossils, because genes preserve traces of ancestry carried continuously from one generation into the next, across hundreds of millions of years. What surprised researchers most was not merely the age of the chimaera lineage, but the unusual stability of its genome. Compared with many other vertebrates, the elephant shark's DNA had changed remarkably little over immense spans of evolutionary time. Mutations accumulated more slowly, large structural rearrangements appeared less common, and many ancestral genetic patterns remained unusually intact. Some scientists began referring to the genome as a molecular time capsule, a surviving archive from an earlier stage in vertebrate evolution preserved with extraordinary fidelity beneath the ocean surface. The sequencing project also revealed what the chimaera lineage had lost. Certain components of the adaptive immune system present in all bony fish, amphibians, reptiles, birds, and mammals were absent entirely. Specific categories of T-cell receptors, essential elements of immune recognition in humans and other tetrapods, simply did not exist within the elephant shark genome. Their absence suggested that these immune structures evolved after the chimera lineage separated from the ancestors of sharks and bony vertebrates. In this way, absence itself became evidence, marking evolutionary innovations that emerged later in the vertebrate story. Yet, the genome also preserved sequences no longer found in sharks, rays, or any living bony vertebrate. Ancient genes, discarded elsewhere, had survived quietly within the chimaera lineage, retained across hundreds of millions of years, while other groups lost them through evolutionary change. These ancestral sequences gave researchers a rare opportunity to approximate what the genome of the earliest jawed vertebrates may have resembled before later branches diverged into the enormous diversity of fish, reptiles, birds, and mammals that now inhabit the planet. Because humans share common ancestry with all vertebrates, these findings extend far beyond the biology of a single deep-sea fish. The developmental systems shaping human teeth, sensory organs, cartilage, and aspects of immune function emerged gradually from structures already present in distant aquatic ancestors. By identifying which genes the chimera retained and which it never acquired, scientists gained a clearer picture of how vertebrate biology assembled itself over geological time. Some of the molecular pathways responsible for mineralizing cartilage in chimeras resemble early stages of bone formation in humans, while other retained sequences illuminate primitive sensory and immune mechanisms from which more complex systems later evolved. The genome sequencing also resolved a long-standing uncertainty regarding the chimera's exact evolutionary position. Fossil evidence had suggested an ancient divergence from sharks, but genetic comparisons provided a far more precise estimate. The lineage separated approximately 420 million years ago, slightly earlier than many paleontologists had predicted from anatomy alone. Long before dinosaurs, before forests spread widely across land, before vertebrate life emerged onto continents, the ancestors of the chimera had already begun evolving along their own independent path beneath ancient seas. Inside every living chimera, that separation remains preserved not only in the shape of the body, but in the structure of the genome itself, a biological memory carried silently through time, while entire branches of vertebrate history appeared and vanished around it. The name ghost shark did not emerge from formal taxonomy or careful scientific classification. It came from appearance, from the strange visual impression left behind when deep-sea cameras first illuminated these animals moving through black water. Many species possess pale skin that appears almost translucent beneath artificial light, their bodies reflecting a muted silver white against the surrounding darkness. Combined with the slow, drifting motion of the fins and the sudden flare of green eyes caught in a camera beam, the effect resembles something less like an ordinary fish and more like a shape briefly materializing out of the ocean itself before dissolving back into shadow. Other names followed similar instincts. Ratfish referred to the long, tapering tail trailing behind the body. Rabbitfish emerged from the blunt face and large forward eyes of certain species. Spookfish captured the same unease carried by ghost shark, the sense that the animal belongs to an older, stranger world, hidden below ordinary human experience. None of these names arose from mythology in the traditional sense. They came instead from first reactions, attempts by fishermen, sailors, and researchers to describe an animal that seemed difficult to compare with anything already familiar. Unlike the great white shark, the giant squid, or the coelacanth, the chimera occupies almost no meaningful place in folklore or maritime legend. Across centuries of seafaring history, most humans simply never encountered it often enough for stories to accumulate. The depths inhabited by chimeras remained inaccessible to ordinary fishing methods for most of recorded history, and specimens brought accidentally to the surface usually arrived damaged, decompressed, or already dead. Without regular encounters, there could be no stable mythology surrounding the animal. Entire cultures developed stories around whales, octopuses, and surface sharks because those creatures entered human life repeatedly. The chimaera remained hidden too far below the reach of memory. There were occasional exceptions. Rare individuals washed ashore after storms or appeared unexpectedly in coastal nets, their anatomy distorted and unfamiliar in the light of the surface world. Historical records from certain fishing communities describe strange fish with wing-like fins, glowing eyes, and tails resembling whips or rats. It is likely that some fragment of older sea monster traditions emerged from these encounters, though the evidence remains impossible to trace with certainty. A decompressed chimaera, lying across wet sand, would have looked profoundly alien to observers who possessed no scientific framework for understanding deep-sea animals. The formal scientific name carries its own older mythology. In ancient Greek tradition, the chimaera was a fire-breathing hybrid creature composed from incompatible animals, part lion, part goat, part serpent. Early naturalists applied the name to the fish because its anatomy appeared similarly assembled from mismatched parts. The broad head, flexible snout, immense eyes, wing-like fins, and tapering tail resisted easy categorization. Even within scientific collections, the animal often looked less like a coherent species and more like a reconstruction pieced together from unrelated fragments. Modern reactions to chimaera footage remain strikingly similar. On the rare underwater videos captured by remotely operated vehicles, viewers frequently respond not with recognition, but with disbelief. The comments attached to these recordings often repeat the same impressions in different forms. Many people describe the animal as looking artificial, computer-generated, or designed rather than evolved. Others compare it to fictional extraterrestrial life rather than terrestrial biology. There is a recurring sense that the proportions and movement violate unconscious expectations about how fish are supposed to appear. That reaction itself reveals something scientifically important. Human perception is shaped by familiarity with modern vertebrate anatomy, particularly the streamlined body plans common among shallow water fish. The chimaera diverged from those patterns so early in evolutionary history that its appearance now triggers a kind of cognitive dissonance. The brain searches for familiar categories and fails to resolve them completely. The animal seems assembled incorrectly only because the body plan emerged long before the forms most humans instinctively recognize became dominant across the oceans. In this way, the ghost shark's uncanniness is not an illusion created by darkness or rarity. It is the visible presence of deep, evolutionary time carried forward into the modern world, preserved inside an animal whose anatomy still reflects ancient experiments in vertebrate design that the rest of life gradually left behind. For centuries, the chimaera existed in science as a motionless thing. Naturalists encountered it only after death, hauled upward in fishing nets from depths no human could reach directly. Specimens arrived pale and damaged from decompression, their soft tissues collapsing under surface pressure before they could be examined properly. They were preserved in alcohol-filled jars, pinned onto dissection tables beneath laboratory lights, or stored in museum collections where their bodies slowly stiffened into anatomical reference material. Researchers could measure fins, classify tooth plates, and compare skeletal structures, but they had never seen a living chimaera move through its own environment. Entire descriptions of behavior had to be inferred from anatomy alone. That began to change during the final decades of the 20th century, when remotely operated vehicles capable of descending below 500 meters became increasingly reliable. Throughout the 1980s and 1990s, advances in underwater cameras, fiber optic communication systems, and deep-sea robotics opened an entirely new way of studying marine life. For the first time, scientists could enter deep water visually without destroying the organisms they wished to observe. The ocean floor ceased to be accessible only through trawl nets and preserved remains. Cameras could now remain submerged for hours, drifting quietly across continental slopes while transmitting moving images back to research vessels above. Early chimera footage captured during these surveys was often brief and technically crude by modern standards. Grainy monochrome silhouettes crossed dim screens for only a few moments before disappearing into darkness again. Yet, even these fragmented recordings carried enormous significance. The movement matched what comparative anatomists had predicted for decades. Chimaeras did not thrash through the water like pelagic sharks. They glided with controlled motions of the pectoral fins, maintaining a calm buoyancy close to the seafloor while the body itself remained relatively still. The old anatomical assumptions had been correct, but seeing the behavior unfold directly transformed the animal from a preserved curiosity into a living participant in its environment. Among the most important recordings came from the Monterey Bay Aquarium Research Institute, known widely as MBARI, whose remotely operated vehicles explored the continental margins off California during the early years of the new century. Their footage of spotted ratfish provided some of the first truly high-quality images of living chimeras in their natural habitat. Illuminated by the soft white beams of the vehicle's lights, the fish moved slowly above the sediment, searching the seafloor with no visible sign of distress. Rather than fleeing the approaching machine, many individuals continued foraging almost undisturbed, their large, reflective eyes catching the light while the fins opened and closed in measured rhythm. The difference between live chimeras and preserved specimens immediately became obvious. In museums, the skin often appears dull, gray, or pale brown, flattened by preservation, fluid, and time. In life, the body carries an iridescent sheen that shifts subtly beneath illumination, almost metallic in places. The eyes flare, with green reflections impossible to reproduce in dead tissue, and the movement itself changes the entire perception of the animal. Preserved specimens suggest awkwardness, but living chimeras possess a fluid grace that photographs alone cannot fully communicate. Another important moment arrived in the year 2016, when MBARI recorded footage near Hawaii of what appeared to be a hydrolagus species not previously documented in that region. The animal moved across the deep Pacific seafloor at considerable depth, its presence raising immediate questions about how widely certain chimera species may actually be distributed. Because deep-sea sampling remains limited across enormous areas of ocean, many assumptions about species ranges are still provisional. A single unexpected sighting can alter scientific understanding of distribution patterns that had remained unchallenged for decades. Even now, however, only a small fraction of known chimera species have ever been filmed alive in their natural environment. Most continue to exist primarily through preserved specimens and scattered trawl records, their behaviors still inferred more often than directly observed. Across the deep ocean, there are species whose movements, courtship, feeding patterns, and daily lives remain completely undocumented by cameras, continuing unseen through darkness where human observation has only just begun to arrive. The deeper water becomes, the heavier it grows. Every descent through the ocean adds pressure from the immense column of seawater above, pressing downward continuously, from all directions at once. Roughly every 10 meters of depth adds another atmosphere of pressure. By 1,000 meters below the surface, a chimaera experiences approximately 100 times the pressure present at sea level, a force so immense that most surface-adapted animals would collapse under it almost immediately. Pressure behaves differently underwater than many people imagine. It does not arrive as impact or crushing force against a single surface. Instead, it surrounds the body completely, compressing every gas-filled cavity and altering the behavior of molecules themselves. Lungs would shrink catastrophically. Swim bladders, the gas-filled organs many fish use to regulate buoyancy, would compress into uselessness long before reaching true deep ocean conditions. Surface animals depend on internal spaces filled with air, but in the abyss those spaces become liabilities. The chimaera avoids one of the greatest problems of deep living simply by lacking a swim bladder entirely. Like sharks and rays, it does not rely on trapped gas to maintain buoyancy. Instead, the body evolved along another path, one more stable under immense pressure. Without compressible gas cavities, there is nothing inside the fish to implode as it descends into the cold depths. Its body remains structurally suited to environments where pressure never relaxes. Neutral buoyancy comes instead from chemistry. Much of the chimaera's lift is generated by a remarkably large liver saturated with low-density oils, particularly a compound called squalene. This oily substance is lighter than seawater and helps offset the natural tendency of the body to sink. Squalene now appears in certain cosmetics and vaccine formulations used by humans, but within the chimaera it serves a far older purpose, allowing the animal to move through the deep ocean without constant muscular effort. In many species, the liver accounts for between 10 and 20% of total body mass, an extraordinary proportion reflecting how central this organ is to survival at depth. Inside the abdominal cavity, it occupies a large volume relative to the rest of the body, functioning less like an accessory digestive structure and more like part of the animal's flotation system itself. Combined with the chimaera's slow movement and energy-efficient anatomy, the buoyant liver allows long periods of gliding through the water with minimal exertion. Adaptation to deep pressure extends far beyond visible anatomy. At the cellular level, the membranes surrounding chimera cells differ chemically from those of shallow water animals. Cell membranes must remain flexible enough to regulate the movement of nutrients, waste, and chemical signals across microscopic boundaries. Under extreme pressure, ordinary membranes become increasingly rigid and lose functionality. Deep-sea organisms compensate by altering the lipid composition of those membranes, preserving fluidity even in cold, high-pressure conditions where surface-adapted cells would malfunction. Proteins face similar challenges. The molecules responsible for nearly every biological process must maintain precise three-dimensional structures in order to function properly. At depth, intense pressure can destabilize or deform proteins evolved for surface conditions, disrupting metabolism and cellular regulation. Deep-sea proteins evolved differently. Their structures fold and stabilize in ways that remain functional under pressures capable of denaturing the proteins of most shallow water species. For this reason, chimaeras and other abyssal organisms have become valuable subjects in molecular biology research. Scientists studying pressure adaptation examine how these proteins behave under conditions that resemble the deep ocean, searching for broader principles about biochemical stability itself. Some of the mechanisms preserving protein function in chimeras may eventually inform biotechnology, medicine, and the understanding of how life survives in extreme environments beyond Earth's surface oceans. Yet within the animal, none of this adaptation appears dramatic. The chimaera simply moves through its environment as though the crushing pressure surrounding it does not exist at all. The body has become so completely integrated with the physical realities of the deep sea that what would destroy most vertebrates became ordinary background conditions long ago, absorbed into the quiet architecture of survival, written gradually into flesh, oil, membranes, and proteins across hundreds of millions of years beneath the weight of the ocean. Below the reach of storms and tides, the ocean separates itself into layers of fading light and growing cold. The chimaera passes through these regions not as a visitor but as a permanent inhabitant, moving across vertical worlds that most surface animals could never endure. The uppermost of these deep layers is the mesopelagic zone, stretching roughly from 200 to 1,000 meters beneath the surface. Oceanographers often call it the twilight zone because sunlight still exists there in weakened form, filtered down into dim blue shadows too faint to sustain photosynthesis. Shapes dissolve quickly into darkness. Distance becomes difficult to judge. In this fading environment, bioluminescence begins replacing sunlight as the dominant form of illumination. Tiny flashes pulse constantly through the water column. Copepods emit brief bursts of light when disturbed. Jellyfish drift with luminous edges glowing softly in the dark. Lanternfish carry rows of photophores beneath their bodies like submerged constellations. The chimaera moves among these intermittent signals with eyes adapted to gather even the faintest trace of available light, navigating a region where day and night have already begun to lose meaning. Deeper still lies the bathypelagic zone, extending from 1,000 to 4,000 meters beneath the sea surface. Here, sunlight disappears entirely, not dimmed, not weakened, but absent. The darkness becomes complete and permanent, interrupted only by the cold blue flickers produced by living organisms themselves. In this environment, there is no horizon, no silhouette against distant light, no dawn arriving from above. Time passes without visible cycles. The ocean exists in continuous night. At these depths, the seafloor spreads outward in broad plains of fine sediment accumulated slowly over immense spans of time. Marine snow settles continuously onto this landscape, forming layers of pale material that can drift undisturbed for centuries. The terrain often appears almost empty at first glance, smooth and gently undulating beneath the lights of descending research vehicles. Yet scattered across this sediment are rocky outcrops, cold seeps leaking mineral-rich fluids from beneath the crust, and the skeletal remains of whale falls where entire temporary ecosystems gather around a single carcass descending from the distant world above. The temperature rarely changes. Most chimaera habitats remain between 2 and 4 degrees Celsius, cold enough to slow metabolism, movement, growth, and decay itself. Biological processes unfold gradually here. Muscles contract more slowly. Digestion requires longer periods. Reproduction stretches across extended timescales, measured not in rapid seasonal cycles, but in patient intervals shaped by scarcity and cold stability. Despite appearances, the deep seafloor is densely alive in quiet ways. Brittle stars spread their thin arms across the sediment, searching for drifting organic particles. Sea cucumbers process the mud itself, extracting nourishment from material too diffuse for larger animals to use efficiently. Polychaete worms tunnel beneath the surface, while isopods move slowly across the plain like armored scavengers. Much of the ecosystem depends on material falling from far above, because almost no energy originates locally in the absence of sunlight. That descending material is known as marine snow, though the name conceals how strange the process truly is. Across the entire ocean, dead plankton, microscopic fecal pellets, fragments of algae, shed exoskeletons, and tiny decomposing particles drift downward continuously through the water column. The fall is slow and constant, a perpetual rain of organic debris sinking from the productive surface into the deep. Every ecosystem below ultimately depends on this steady downward transfer of energy. The chimaera's prey feeds upon it, and the chimaera in turn feeds upon them, linked invisibly to distant sunlight it will never encounter directly. From the perspective of the chimera itself, this environment is defined less by sight than by subtle sensation. Water pressure presses from all directions without relief. Near freezing currents move gently across the body. Tiny vibrations travel through the sediment when another organism shifts nearby. Chemical traces drift invisibly through the water. Particles settle slowly from above through darkness, so complete that vision alone becomes insufficient for survival. And through all of it, across 400 million years of planetary change, the chimaera has continued navigating this cold, unseen world with almost no interruption, moving through an environment that remains among the least familiar places on Earth to the species living far above it. Time passes strangely in the deep ocean. Growth slows, movement slows, even decay unfolds with unusual patience beneath the cold pressure of the abyss. For the chimaera, this slow environment makes one of the simplest biological questions unexpectedly difficult to answer. Scientists still do not know with confidence how long most species actually live. Among all the unresolved mysteries surrounding the group, lifespan remains one of the largest and most persistent gaps in understanding. Determining age in cartilaginous fish is already challenging under ordinary circumstances. Unlike many bony fish, which possess calcified ear structures called otoliths that preserve annual growth layers clearly, sharks and chimaeras leave behind subtler records of time. Researchers instead examine calcified regions within the body, particularly the vertebral centra or dorsal spines, searching for concentric growth bands, similar in appearance to tree rings. In theory, each band reflects a period of slowed or accelerated growth linked to seasonal changes in metabolism and food availability. For chimaeras, the dorsal spine positioned before the first fin became the primary structure used for age estimation. Thin sections of the spine are cut and examined under magnification, revealing alternating layers of denser and lighter material deposited gradually through life, yet interpreting these patterns remains uncertain. Deep-sea environments often lack the strong seasonal cycles present in shallower ecosystems, and food availability may fluctuate irregularly over time. Growth does not always proceed at a predictable rate. Some rings may represent annual cycles, while others may form under entirely different physiological conditions. Two researchers studying the same spine can arrive at different estimates for the same individual. Current evidence suggests that many chimera species are relatively long-lived for animals of their size. Existing estimates commonly range between 15 and 30 years, though most remain poorly validated and based on limited sample sizes. These values may ultimately prove accurate for some shallow water species, but deep-sea chimeras likely live under biological conditions very different from those nearer the surface. Their slow metabolism, delayed reproduction, and cold, stable habitat all point toward longer lifespans than researchers can currently verify directly. Comparisons with other deep-water cartilaginous fish hint at possibilities far beyond those early estimates. The Greenland shark, another cold-adapted species inhabiting dark northern waters, transformed scientific assumptions about fish longevity when radiocarbon analysis of proteins inside the eye lens suggested lifespans exceeding 400 years. Some individuals alive today may have already existed before the beginning of the industrial era. That discovery forced marine biologists to reconsider how slowly life can unfold in the deep ocean. The comparison naturally raises similar questions about chimaeras. If another deep-dwelling cartilaginous fish can survive for centuries in cold, high-pressure environments, it becomes difficult to dismiss the possibility that certain chimera species may also possess extraordinary longevity. Their physiology shares many of the same broad characteristics associated with slow aging, reduced metabolic demand, and stable environmental conditions. Yet unlike the Greenland shark, no equivalent radiocarbon aging study has ever been completed for chimeras. Part of the difficulty lies in obtaining appropriate material for analysis. Deep-sea species are rarely encountered alive, and known age individuals do not exist for long-term comparison studies. Even when specimens are collected, preservation methods may damage tissues needed for precise chemical analysis. Building the kind of dataset required for reliable lifespan estimates becomes extraordinarily difficult when the animals themselves remain inaccessible across most of their range. What scientists suspect, however, carries important consequences regardless of the exact numbers involved. Species that grow slowly, mature late, and reproduce gradually recover poorly from population decline. A fish that requires decades to replace itself cannot withstand intense harvesting pressure in the way fast-breeding coastal species sometimes can. This vulnerability already defines many deep-sea organisms whose evolutionary strategy depends not on rapid reproduction, but on persistence through stability and patience. The chimera appears shaped by that same ancient rhythm. Everything about its biology suggests a life unfolding gradually in cold darkness, measured not by urgency or rapid change, but by long, uninterrupted continuity beneath the ocean floor, where time itself seems to move more slowly than it does in the bright, restless waters above. For most of human history, the chimera escaped industrial exploitation simply because it remained inconvenient. It lived too deep to capture easily, reproduced too slowly to sustain heavy harvest, and possessed an anatomy unfamiliar enough that few commercial markets ever developed around it. Unlike tuna, cod, or surface sharks, chimeras never became central to global fishing economies. Their flesh was rarely sought deliberately, and many species inhabited depths beyond the routine reach of earlier fishing technology. In a strange way, obscurity protected them. There were exceptions. During the 19th and early 20th centuries, certain fisheries targeted chimeras for the oil contained within their enlarged livers. Rich in squalene and other low-density lipids, the oil proved useful as a lubricant for delicate precision instruments, including watches, scientific equipment, and firearms. In some coastal regions, this trade expanded enough to produce local declines in populations, particularly where shallow water species could be accessed more consistently. The extraction remained relatively small compared with industrial whaling or cod fisheries, yet it revealed how quickly even obscure deep-sea animals could become vulnerable once a commercial use emerged. Today, the primary threat arrives less directly. Most chimeras are not intentionally pursued at all. Instead, they die as bycatch in deep-sea trawl fisheries, targeting entirely different species, particularly orange, ruffy, grenadiers, and deep-water sharks. Massive weighted nets drag across the seabed, collecting whatever organisms occupy the same habitat. Chimeras caught in these operations are usually discarded after capture, often already dead or too badly damaged to survive decompression and handling. The problem is compounded by the biology of the animals themselves. Chimaeras reproduce slowly, producing relatively few offspring over long intervals. Many species deposit only 1 or 2 embryos within each leathery egg capsule, and incubation may continue for months before hatching occurs. Evidence also suggests that sexual maturity arrives late in life, though precise ages remain uncertain for most species. In ecological terms, this combination creates a lineage poorly suited to recovering from sudden population losses. Where faster reproducing fish may rebound after exploitation, deep-sea chimeras likely require decades to restore depleted numbers. Yet, measuring those declines remains extraordinarily difficult because basic population data barely exists. Across the majority of species, scientists cannot estimate abundance with meaningful precision. The International Union for Conservation of Nature, known as the IUCN, lists many chimeras as data deficient. The designation does not mean the animals are safe. It means there is insufficient information even to determine whether they are threatened at all. For a vertebrate lineage more than 400 million years old, the lack of knowledge itself becomes part of the story. This absence of data reflects how recently deep-sea ecosystems entered serious scientific focus. Trawl records remain patchy, research surveys cover only limited regions, and most species are encountered so infrequently that long-term population trends become nearly impossible to calculate. A species could decline substantially across large portions of its range before enough information accumulates for researchers to recognize the pattern with confidence. One unusual case stands apart from the rest. In southern Australia and New Zealand, Cartilaginous milli supports a managed commercial fishery. Sold commonly under the market name flake, the fish appears in some fish and chip shops where consumers may never realize they are eating a chimera at all. The neutral trade name obscures the animal's identity, separating the product from the strange, deep evolutionary history carried within its body. Compared with many deep-sea relatives, the elephant shark inhabits shallower coastal waters where commercial harvest becomes economically feasible. Even there, however, sustainable management remains difficult without precise biological information. Reliable age estimates, reproductive rates, migration patterns, and population sizes remain incomplete. Fisheries management depends upon understanding how quickly a species can replace itself after harvest, but with chimeras, many of those calculations rest on uncertain foundations. The broader reality is uncomfortably simple. Humanity has developed the technological ability to reach deep ocean ecosystems far more quickly than it has developed the scientific understanding necessary to manage them responsibly. Nets now descend routinely into habitats that remained physically inaccessible for most of history, collecting organisms whose lifespans, reproductive cycles, and population structures remain largely unknown. Somewhere beyond the reach of those trawl lines, chimeras continue moving slowly across the seafloor as they always have, but the isolation that protected them for millions of years no longer guarantees protection in the modern ocean. The history of the chimera survives only in fragments. Across 400 million years of existence, the physical evidence left behind by the lineage occupies a surprisingly small amount of space, scattered through museum collections, in drawers, filled not with complete skeletons, but with isolated teeth, broken spines, and occasional impressions pressed into ancient stone. Because the chimera's body is composed primarily of cartilage rather than bone, most of it disappears, long before fossilization can occur. Soft cartilage decays quickly after death and rarely survives the conditions necessary for preservation. Entire populations may have lived and vanished without leaving behind more than a few mineralized traces. What does preserve are the hardest structures. Tooth plates endure particularly well because they are heavily mineralized and resistant to decay. Dorsal spines survive more rarely, but still appear in ancient sediments with enough consistency to map the lineage through deep geological time. These remains have been recovered from Devonian, Carboniferous, Permian, Triassic, Jurassic, Cretaceous, and Paleogene rock formations spread across every continent. The fragments appear and reappear through Earth's history like faint recurring signals, enough to prove continuity but rarely enough to reconstruct a complete living animal. Among the most important early groups are the Iniopterygians, small, deep-bodied relatives that flourished during the Carboniferous period between roughly 360 and 300 million years ago. Their fossils provide one of the clearest glimpses into the early diversification of holocephalans, the broader group that includes all living chimaeras. Iniopterygians possessed unusual body proportions, broad fins, and cranial structures unlike anything now alive. Some species appear almost experimental in their anatomy, reflecting an ancient phase of vertebrate evolution when body plans remained more fluid and diverse than those surviving today. Another significant group, the cochleodontids, is known primarily from distinctive coiled tooth plates preserved in Paleozoic rocks. The spiraling mineralized structures look almost architectural when isolated from the rest of the animal, their grinding surfaces adapted for crushing hard-shelled prey. In many cases, the tooth plates are all that remain. The body itself vanished hundreds of millions of years ago, leaving paleontologists to infer the appearance and ecology of the animal from feeding structures alone. Rarely, preservation becomes extraordinary. In limestone deposits from Montana dating back approximately 330 million years, fossils of Balanci and Netsapoi retained impressions of soft tissues alongside skeletal structures. These specimens remain among the most important holocephalan fossils ever discovered because they preserve outlines of the body itself rather than isolated fragments. Fins, body contours, and cranial proportions survived within limestone nodules that sealed the animals rapidly after death. For paleontologists accustomed to reconstructing chimaera relatives from scattered remains, these fossils offered an unusually complete glimpse into ancient forms moving through Carboniferous seas. What they revealed was striking diversity. Some ancient relatives possessed elaborate cranial crests extending from the head in shapes absent from living species. Others reached lengths approaching 3 meters, far larger than most modern chimeras. Certain forms appear to have inhabited shallow reef ecosystems rather than the cold, deep water environments favored today. The ancient holocephalans were not always quiet specialists of the abyss. At times they occupied a broad range of ecological roles across marine ecosystems that no longer exist. Somewhere between those ancient seas and the modern ocean, that diversity narrowed dramatically. The transition from the varied body plans of Paleozoic holocephalans to the relatively conservative anatomy of living chimeras represents a major evolutionary bottleneck. Many paleontologists suspect the Permian-Triassic extinction event played a central role in this contraction. Around 252 million years ago, ecosystems across the planet collapsed under volcanic-driven climate catastrophe, ocean acidification, and widespread oxygen depletion. Entire marine lineages disappeared permanently. The surviving chimaeras may represent only a small remnant of what the group once was. The approximately 50 living species known today could be the final branches of a much larger evolutionary radiation that previously filled oceans with forms now erased almost completely from the fossil record. Their strange anatomy, once perhaps only one variation among many, became the surviving template carried forward after countless sister lineages vanished into extinction, leaving behind little more than teeth, spines, and faint impressions sealed inside ancient stone. Long before the asteroid that ended the age of dinosaurs, Earth passed through a far greater catastrophe. Around 252 million years ago, near the boundary between the Permian and Triassic periods, the planet experienced the most severe mass extinction known in the geological record. Approximately 96% of all marine species disappeared. On land, roughly 70% of vertebrate species vanished as ecosystems collapsed across continents and oceans alike. Entire branches of evolutionary history ended permanently during this interval, leaving behind a world profoundly emptier than the one that existed before it. The disaster unfolded over immense spans of time rather than a single sudden moment. In what is now Siberia, volcanic eruptions on a scale almost impossible to comprehend began pouring lava across the landscape in repeated pulses lasting hundreds of thousands of years. These eruptions, now known collectively as the Siberian Traps, released enormous quantities of carbon dioxide and sulfur dioxide into the atmosphere. Global temperatures rose rapidly, ocean chemistry shifted, rainfall patterns changed across continents. The atmosphere itself became unstable under the weight of volcanic gases accumulating faster than Earth's systems could absorb them. The oceans suffered especially severe consequences. As carbon dioxide dissolved into seawater, acidification intensified, altering the chemistry required for many organisms to build shells and skeletons. Warming temperatures reduced the amount of oxygen seawater could retain, while circulation patterns weakened across large ocean basins. Vast regions became oxygen-depleted, dead zones where marine animals suffocated slowly in water, no longer capable of supporting normal respiration. During the worst phases of the extinction, much of the ocean transformed into an environment unlike anything familiar in the modern world. Waters grew warmer, more acidic, and increasingly stagnant. Productive ecosystems that had supported complex marine food webs for millions of years deteriorated into biologically impoverished regions where few organisms could survive. Sediment records from the period preserve evidence of widespread anoxia spreading through shallow seas and deeper basins alike, creating some of the harshest marine conditions in Earth's history. Many ancient groups disappeared completely. Trilobites, which had already survived earlier extinctions across hundreds of millions of years, vanished forever during the Permian collapse. Reef-building organisms suffered catastrophic losses that erased entire ecosystems. Numerous ammonite lineages disappeared. Large portions of the fish fauna inhabiting Paleozoic seas failed to cross the extinction boundary into the Triassic world that followed. And yet the ancestors of the chimaera endured. Exactly how they survived remains uncertain. Paleontologists and marine biologists continue debating the mechanisms that allowed a small fraction of holocephalan lineages to persist while so many others vanished permanently. The honest answer is that no single explanation has fully resolved the mystery. One leading possibility involves depth itself. Deep water habitats may have buffered certain populations from the most extreme conditions affecting surface ecosystems. Although oxygen depletion reached many regions of the ocean, some deeper environments may have remained comparatively stable in temperature and chemistry. Organisms already adapted to cold, dark habitats could have avoided portions of the environmental chaos, devastating shallower marine communities. Metabolism may also have played an important role. Chimeras possess exceptionally slow biological rhythms compared with many active predatory fish. A low metabolic rate reduces oxygen demand, potentially allowing survival in waters where faster metabolizing animals could no longer function effectively. In a world where marine oxygen levels collapsed across enormous regions, simply requiring less oxygen may have become a decisive evolutionary advantage. Dietary flexibility may have contributed as well. Chimeras feed on a wide variety of benthic invertebrates and scavenged material rather than depending exclusively on fast-moving prey or highly specialized food sources. As ecosystems deteriorated and species disappeared, this broader feeding strategy may have allowed surviving populations to shift gradually between available food sources while more specialized animals starved. Survival, however, did not mean escape without loss. The fossil record suggests holocephalan diversity declined dramatically across the Permian boundary. Many ancient forms disappeared entirely, including numerous body plans and ecological strategies that had flourished during earlier geological periods. Only a relatively small number of lineages appear to have persisted into the Triassic, carrying forward the ancestry that would eventually produce all living chimeras. The ghost shark lineage, emerging from the extinction, was therefore not the untouched continuation of an ancient world, but the reduced remnant of one. It survived the greatest biological collapse Earth has ever experienced, though much of its broader evolutionary family did not survive alongside it. Far above the deep ocean floor, in a world still dominated by giant reptiles, the final days of the Cretaceous period arrived with extraordinary suddenness. Around 66 million years ago, an asteroid estimated at roughly 10 to 15 kilometers wide entered Earth's atmosphere at cosmic speed and struck the region now known as the Yucatan Peninsula in Mexico. The impact carved a crater more than 180 kilometers across and released an amount of energy equivalent to billions of nuclear warheads detonating almost simultaneously. For a brief moment, the atmosphere itself became incandescent. Rock at the impact site vaporized instantly. Molten debris blasted upward beyond the atmosphere before falling back across the planet as superheated ejecta, igniting wildfires across entire continents. Tsunamis radiated through ancient seas. Shockwaves circled the globe repeatedly. Soot, ash, and pulverized rock accumulated in the upper atmosphere until sunlight reaching the surface diminished catastrophically. The event marked the beginning of the Cretaceous-Paleogene extinction, one of the great biological turning points in Earth's history. Photosynthesis collapsed across large portions of the planet as darkness and cooling spread through the atmosphere. Food chains dependent upon surface productivity began to unravel. Forests disappeared in many regions. Marine ecosystems suffered immense losses as plankton populations declined and the biological foundation of the ocean weakened. Approximately 75% of all species vanished during this interval. Every non-avian dinosaur lineage disappeared permanently. The giant mosasaurs that once dominated marine ecosystems vanished from the oceans. Plesiosaurs, which had survived since the Triassic period, disappeared alongside them. Numerous ammonite species, survivors of multiple earlier extinctions, finally reached the end of their evolutionary history here. Yet even during this catastrophe, the deep ocean remained partially insulated from the immediate violence unfolding above. Cold abyssal water changes slowly. Unlike surface environments directly exposed to atmospheric collapse and wildfire-heated skies, the deep seafloor existed beneath immense layers of water possessing extraordinary thermal inertia. Darkness already defined that world long before the asteroid arrived. Temperatures remained cold and stable, pressure remained constant. The collapse of surface productivity eventually propagated downward through the food web, reducing the slow rain of organic matter descending into the abyss. But these effects unfolded gradually rather than instantaneously. For organisms already adapted to scarcity, cold, and darkness, the deep sea may have functioned as one of the planet's last relatively stable refuges during the extinction. The chimaera lineage endured this catastrophe as well. Fossil tooth plates and dorsal spines identified from geological layers on both sides of the Cretaceous-Paleogene boundary confirm that holocephalan lineages persisted through the extinction without any obvious large-scale collapse. While many marine reptile groups disappeared permanently, the deep-water descendants of ancient Devonian fish continued moving quietly across the seafloor, almost unchanged in their fundamental biology. There is something difficult for the human mind to fully absorb in that comparison. The asteroid impact that ended the age of dinosaurs altered the trajectory of life on land so profoundly that mammals eventually inherited ecological opportunities that had previously remained closed to them. From those surviving mammal lineages came primates, then humans, then ships, laboratories, satellites, and remotely operated vehicles descending at last into the deep ocean to film the ghost shark moving through darkness. And through all of that time, the chimaera remained. It outlasted the placoderms that once ruled Devonian seas. It survived beyond the trilobites that had inhabited Earth long before vertebrates became dominant. It endured after the extinction of mosasaurs, plesiosaurs, and the non-avian dinosaurs themselves. Entire dynasties of dominant vertebrate life rose, expanded across the planet, and disappeared, while the chimaera lineage persisted in cold, deep water beneath them all. The deep ocean remembers evolutionary time differently from the surface world. In that darkness, catastrophe arrives muted and delayed, filtered through kilometers of water and years of drifting organic debris. The ghost shark passed through the asteroid winter, not because it conquered the extinction, but because its world existed slightly apart from the one collapsing above. Along the cold coastal shelves of southern Australia and New Zealand lives a chimaera, unlike most of its deep water relatives. While ghost sharks farther offshore remain hidden beneath kilometers of dark ocean, this species rises into comparatively shallow seas where human researchers can occasionally reach it alive. Cartilaginous millii, more commonly known as the elephant shark, has gradually become the single most important window into the biology of an entire ancient lineage. Its body still carries the unmistakable silhouette of a chimaera, soft cartilaginous frame, wing-like pectoral fins, an elongated tail tapering into darkness behind it. Yet the face is what first captures attention. Extending from the snout is a flexible projection ending in a fleshy, rounded disc, almost like a delicate trunk. This strange structure bends slowly through the sediment as the animal moves across the seafloor, probing mud and sand for buried invertebrates. In fully grown adults, the body may reach roughly 1.2 meters in length, though much of that length belongs to the trailing tail itself. Unlike deeper species adapted to intense pressure and near-freezing abyssal water, the elephant shark survives long enough in controlled aquarium conditions for scientists to study it directly. This difference has shaped nearly everything modern biology knows about chimaeras. Deep-sea species hauled rapidly to the surface usually die before meaningful observation becomes possible. Their bodies are adapted to stable darkness, immense pressure, and extremely specific environmental chemistry. The elephant shark alone has proven resilient enough to bridge the distance between abyssal biology and laboratory science. Inside research facilities, Cartilaginous has revealed a physiology both ancient and unexpectedly elegant. Studies of its immune system showed that it possesses a simplified but fully functional adaptive immune response. Some of the complex molecular machinery found in bony vertebrates is absent, yet the system still performs effectively enough for survival. These findings helped clarify which portions of vertebrate immunity evolved early in jawed fish ancestry and which appeared later in evolutionary history. Other research focused upon the proteins within its tissues, particularly proteins capable of remaining stable under environmental conditions that would damage the cellular machinery of many surface-adapted organisms. Scientists examining these molecular structures found clues about how ancient cartilaginous vertebrates maintained functional biology across cold marine environments for immense spans of geological time. Even the composition of cartilage itself has become scientifically valuable, offering insight into the earliest stages of vertebrate skeletal evolution. Some of the most remarkable observations emerged from reproductive studies. Chimaera eggs develop inside elongated leathery capsules deposited upon the seafloor, and in Cartilaginous portions of these capsules are translucent enough for researchers to observe embryonic growth directly. Within those transparent windows, tiny vertebrate bodies slowly assemble themselves according to developmental instructions inherited from ancestors that diverged from the rest of the jawed vertebrates roughly 420 million years ago. The embryos develop gradually, over periods extending many months. Fins emerge first as delicate folds along the body. Sensory organs take shape slowly beneath the thin membrane of the egg capsule. The jaws harden. The cartilaginous skeleton expands into place. Watching this process unfold gives developmental biologists something extraordinarily rare: a living glimpse into patterns of vertebrate construction rooted deep within evolutionary history itself. Comparisons between chimaera embryos and embryos of sharks, bony fish, amphibians, reptiles, birds, and mammals helped scientists separate ancient developmental mechanisms from newer evolutionary innovations. Some tissue formation pathways appear almost universal across jawed vertebrates, preserved despite hundreds of millions of years of divergence. Others belong only to later branches of the vertebrate tree, additions layered gradually upon older biological foundations. Yet even now, uncertainty surrounds every conclusion. Everything learned from Cartilaginous milii comes from a single, relatively accessible species living near coastlines rather than within the deepest abyssal habitats occupied by many other chimaeras. The elephant shark may possess behaviors, metabolic traits, or physiological tolerances unlike those of its distant relatives below. In many ways, the laboratory model remains only one illuminated fragment of a lineage still largely hidden in darkness. For much of the 20th century, scientists quietly assumed the chimaera lineage was essentially complete from a taxonomic perspective. The known species were few in number, rarely encountered, and distributed through remote regions of the deep ocean where systematic biological surveys remained difficult and expensive. Compared to insects, reef fish, or tropical amphibians, the group appeared almost static, a small surviving branch of vertebrate history already mapped and cataloged. Yet even now, new ghost sharks continue emerging from the dark. Most discoveries begin not with deliberate exploration, but with accident. a deep-sea trawl targeting commercially valuable fish, drags across the seafloor thousands of meters below the surface. Amid damaged grenadiers, crustaceans, and fragments of sediment lies an unfamiliar specimen, usually dead before it reaches daylight. The animal is preserved, labeled, and eventually transported to a museum collection or university laboratory where it may remain for months or years before a specialist examines it closely. The process of recognizing a new species unfolds slowly and carefully. Taxonomists compare measurements against existing descriptions archived across decades of scientific literature. The proportions of the pectoral fins are examined. The shape and mineral structure of the tooth plates are compared under magnification. Snout length, dorsal spine morphology, coloration patterns preserved in tissue, and the arrangement of sensory canals across the head are all evaluated for subtle distinctions that might separate one lineage from another. Sometimes, those differences prove meaningful. In 2009, researchers formally described Hydrolagus macoscari from specimens collected near the Galapagos Islands. The species possessed anatomical traits distinct enough to separate it from previously recognized hydrolagus forms known elsewhere in the Pacific. Several years later, in 2016, Chimaera opalescens was described from the Mediterranean and northeastern Atlantic. Its pale iridescent appearance and body proportions distinguishing it from its closest relatives. Throughout the 2010s, additional hydrolagus species emerged gradually from Pacific deep-sea surveys, each one revealing that the apparent simplicity of the chimaera family concealed greater diversity than earlier generations suspected. Increasingly, anatomy alone is no longer considered sufficient. Molecular analysis now plays a central role in confirming whether unusual specimens represent truly distinct species or merely regional variations within already described populations. Small tissue samples preserved in ethanol can be sequenced and compared genetically against specimens housed in museum collections across the world. DNA analysis sometimes confirms visible differences. In other cases, animals that appear nearly identical externally reveal deep genetic separation accumulated over millions of years of isolation in different ocean basins. This work continues quietly in laboratories and museum archives scattered across multiple continents. Drawers containing specimens collected decades ago are being reopened and re-examined with tools unavailable to earlier researchers. Some preserved chimeras gathered during forgotten trawl expeditions may already represent undescribed species waiting unnoticed in storage cabinets. The implications stretch outward into the geography of the deep ocean itself. Large portions of the abyssal world remain biologically unexplored. The deep Indian Ocean, vast regions of the southern Pacific, and countless remote continental slopes have never been systematically surveyed for chimeras at all. Even more mysterious are the seamounts rising from the ocean floor. These underwater mountains number in the tens of thousands globally, scattered through darkness like isolated islands separated by immense distances. Ocean currents interacting with their slopes create local ecological conditions capable of supporting highly specialized communities found nowhere else. Most seamounts have never been visited by remotely operated vehicles. No human eyes have directly observed the life moving across their sediment or sheltering among their rocky outcrops. Each one may contain lineages entirely absent from known scientific records. For this reason, some taxonomists now suspect the true number of living chimaera species may approach 70 or 80 rather than the roughly 50 currently described. If correct, nearly half the lineage would still remain unnamed by science, despite 4 centuries of formal zoological study. The ghost shark persists not only through geological time, but through scientific uncertainty itself. Even after centuries of collecting, sequencing, dissecting, and classifying, the deep ocean continues revealing that the ancient lineage beneath its darkness is larger and stranger than humans first believed. Across the surface of the chimaera's snout, almost invisible beneath the pale skin and drifting sediment of the deep ocean, lies another sensory system that still remains only partially understood. These openings appear at first glance like tiny imperfections in the flesh, small pores scattered across the front of the head in subtle geometric patterns. In reality, they form part of one of the strangest structures in cartilaginous fish biology, the rostral organ. Unlike the ampullae of Lorenzini found in sharks and rays, the chimaera's sensory system follows a different anatomical design entirely. The pores covering the snout are filled with conductive mucus and connected internally to branching canals embedded within the cartilage of the head. At the ends of these canals sit specialized sensory cells capable of responding to changes in the surrounding environment. From the outside, the arrangement looks delicate and understated, yet beneath the skin exists an intricate biological network shaped by hundreds of millions of years in darkness. Scientists believe the rostral organ functions primarily as a system for detecting extremely subtle disturbances in the water and sediment surrounding the animal. A crab shifting beneath the seafloor produces tiny vibrations. A buried worm contracts its muscles within the mud. Even the faint electrical activity generated by living tissue may spread weakly through conductive seawater. Somewhere above this hidden movement, the chimaera drifts slowly over the sediment, its snout suspended just above the ocean floor, like an instrument listening to the earth itself. The exact capabilities of this system remain uncertain. Anatomically, the sensory cells inside the rostral organ resemble electroreceptors known from sharks, but they are not identical. Their structure differs in enough ways that many researchers believe the two systems evolved independently after the shark and chimaera lineages separated hundreds of millions of years ago. If correct, this would represent another example of convergent evolution among cartilaginous fish, separate evolutionary paths arriving at broadly similar biological solutions for surviving in dark marine environments. The possibility is remarkable because it suggests that electrical sensing may be such an effective adaptation for life beneath the surface that evolution discovered versions of it more than once within related groups. Yet, despite the scientific importance of the question, rigorous experimental evidence remains surprisingly scarce. Most deep-water chimera species die rapidly when brought to the surface, making controlled laboratory testing almost impossible. Electroreception experiments require living animals placed within carefully monitored electrical environments where responses can be measured precisely. Such work has only been feasible with the elephant shark, Cartilaginous milli, and even then, researchers cannot assume that shallow coastal species possess identical sensory abilities to their deep-sea relatives. As a result, much of what is said about the rostral organ remains grounded in informed inference rather than direct proof. Still, anatomy itself provides powerful clues. The density and arrangement of sensory pores across the snout suggest a structure designed for extraordinary sensitivity. If the organ truly detects weak electrical signals, then a chimera moving across the abyssal seafloor may be capable of perceiving the muscular contractions or heartbeats of prey animals buried several centimeters beneath the sediment. In the featureless darkness of the deep ocean, Where vision alone cannot reliably reveal hidden food, such an ability would transform the surrounding seafloor into a map of invisible biological activity. A sea urchin, half buried in marine snow. A crustacean concealed beneath soft mud. A worm shifting slowly through sediment, laid down over thousands of years. The chimaera may sense these animals long before physical contact occurs. There is something quietly revealing in how incomplete this understanding remains. The uncertainty surrounding the rostral organ does not exist because scientists fully investigated the question and failed to reach agreement. In many cases, the question has barely been explored at all. Entire sensory worlds may still operate within the deep ocean beyond the limits of direct observation, carried inside animals whose biology remains largely hidden from human study. Even now, after centuries of marine science and decades of deep-sea exploration, the ghost shark continues, moving through darkness, with systems humans can describe anatomically, yet still scarcely comprehend. There is a peculiar honesty that emerges when scientists speak carefully about chimeras. After centuries of marine biology, fossil analysis, genetics, and deep-sea exploration, the most accurate description of this lineage is still shaped as much by absence as by knowledge. For animals that have existed for roughly 400 million years, the catalog of unanswered questions remains astonishingly large. In many areas of their biology, science is standing not at the edge of certainty, but at the beginning of inquiry itself. The most basic unknown is population size. No researcher can currently state with confidence how many individuals exist within a single chimaera species. There are no reliable global counts, no verified long-term population trends, and in many cases, not even rough regional estimates. For numerous species, the only evidence of their continued existence comes from occasional trawl bycatch or isolated deep-sea footage, separated by years and thousands of kilometers. Entire populations may persist beneath the ocean in numbers that remain completely invisible to human observation. Even their reproductive behavior remains hidden. Somewhere in the deep ocean, chimaeras locate one another in absolute darkness and reproduce successfully enough to maintain lineages older than the dinosaurs themselves. Yet the mechanism by which they find mates remains unknown. Scientists do not know whether they rely primarily upon chemical signals drifting through seawater, pressure waves detected through the lateral line system, electrical sensing through the rostral organ, or some sensory process not yet recognized. No confirmed observation of natural courtship behavior has ever fully resolved the question. The silence surrounding their sensory world extends further still. No formal study examining hearing in chimeras has ever been comprehensively published. Researchers do not know whether these fish detect sound in ways comparable to sharks or bony fish. They do not know whether chimeras themselves produce acoustic signals beneath the surface. The deep ocean is filled with biological sound, low-frequency vibrations generated by whales, fish, crustaceans, tectonic movement, and distant storms crossing the sea above. Yet, the role of sound within chimera behavior remains almost entirely unexplored. Their social behavior is equally uncertain. Most encounters with chimeras involve solitary individuals drifting slowly across the seafloor. Yet deep-sea trawl operations occasionally recover multiple specimens from the same region at the same time. This could indicate temporary aggregations, overlapping habitat preferences, seasonal reproductive gathering, or nothing socially meaningful at all. The available observations are too fragmentary to distinguish between these possibilities. Scientists cannot currently say whether chimeras spend their lives entirely alone or participate in forms of interaction never directly observed by human eyes. Even the length of a chimera's life remains unresolved. Age estimates, based upon growth rings in dorsal spines, provide only tentative approximations, complicated by inconsistent environmental conditions and uncertain annual growth patterns. No wild chimaera individual has ever been reliably aged using the kind of radiocarbon analysis applied successfully to Greenland sharks. It remains entirely possible that some deep water species live far longer than current estimates suggest. At present, nobody truly knows. The venom carried within the dorsal spine also remains largely mysterious. Only 1 or 2 species have undergone formal chemical analysis of venom components, and even those studies remain incomplete. Researchers understand that the spine can inflict severe pain in humans, accompanied in some cases by swelling and systemic symptoms. But the precise molecular mechanisms responsible for these effects are still poorly characterized. The venom system exists as another fragmentary outline rather than a fully illuminated biological process. What makes this ignorance remarkable is not merely the number of unanswered questions, but the scale of the animals involved. Chimaeras are not microscopic organisms hidden in inaccessible caves or obscure insects confined to remote forests. They are a major surviving branch of the vertebrate tree, descendants of one of the earliest jawed fish lineages on Earth. They have persisted through multiple mass extinctions, inhabited every major ocean basin, and survived for hundreds of millions of years alongside changing continents and collapsing ecosystems. Yet until very recently, the deep ocean concealed them almost completely from human curiosity. Most vertebrate groups entered mythology, folklore, hunting traditions, agriculture, or direct human observation thousands of years ago. The ghost shark remained below, moving silently through darkness so complete that even now, in the age of satellites and genome sequencing, its life is still largely unknown. Long before human beings ever saw a living chimaera moving through its natural world, the deep ocean itself acted as a kind of permanent barrier between observer and animal. The trawl net changed that partially, dragging broken glimpses of deep life upward from darkness. But even then, the chimaera remained frozen in stillness, preserved in alcohol jars or stretched beneath laboratory lights. Only recently has technology begun to close the distance between the surface world and the immense cold terrain where these animals actually live. The modern deep-sea remotely operated vehicle, or ROV, represents the greatest shift in chimaera observation since the first deep trawls of the 19th century. These machines descend where human divers cannot survive, carrying cameras, lights, sensors, and robotic arms into regions of the ocean that remained visually inaccessible for most of scientific history. Some modern research ROVs are rated for depths beyond 6,000 meters, capable of reaching abyssal plains where pressure rises to levels that would instantly crush surface equipment, not specifically engineered for such conditions. The cameras mounted on these systems have transformed what scientists can see. Early deep-sea footage often appeared grainy and distant, shapes drifting through darkness with little detail preserved. Modern high-definition imaging now reveals the reflective sheen of chimaera skin, the faint movement of pectoral fins, the texture of sediment disturbed beneath the snout while feeding. In low-light conditions where human vision would fail almost immediately, these camera systems gather enough illumination to reconstruct entire sections of seafloor with remarkable clarity. Manipulator arms attached to the vehicles provide another important change. Rather than destroying animals through indiscriminate trawling, researchers can sometimes collect specimens gently and selectively, preserving delicate anatomical structures that would otherwise be damaged. Precision acoustic positioning systems allow operators aboard research vessels to guide ROVs slowly across underwater terrain with extraordinary control, holding position within meters above the seafloor, even in darkness and shifting currents. Several research programs have become especially important in expanding chimaera observations. The Monterey Bay Aquarium Research Institute, often known as MBARI, has documented deep-sea life for decades, using the ROVs Ventana and Doc Ricketts along the California margin. Their footage provided some of the clearest early observations of living spotted ratfish gliding undisturbed above the sediment. Farther across the Pacific and Indian Oceans, the Schmidt Ocean Institute's ROV Sebastian has recorded additional deep-sea species during long-duration expeditions around seamounts and unexplored ridges. Around New Zealand, surveys conducted by the National Institute of Water and Atmospheric Research, known as NIWA, continue to expand knowledge of Southern Hemisphere chimaeras inhabiting continental slopes and deep basins. At the same time, entirely new methods are emerging that do not require direct observation at all. Environmental DNA, usually shortened to eDNA, has become one of the most promising tools in deep ocean biology. Every marine organism constantly sheds microscopic traces of genetic material into surrounding seawater through mucus, waste, skin cells, and decomposing tissue. By collecting water samples at depth and filtering them carefully, scientists can search for these suspended genetic fragments and identify species that may have passed through the region recently, even if no animal was ever photographed or captured. For elusive deep-sea fish like chimaeras, this approach may eventually reveal distributions far broader than current records suggest. Researchers are also beginning to experiment with miniature acoustic tags capable of recording depth, temperature, and movement patterns in deep water fish. The challenge with chimaeras lies in attachment. Their soft bodies and fragile physiology make long-term tagging technically difficult, especially for species that cannot tolerate rapid ascent to the surface. Another valuable tool is the baited camera lander, a weighted frame carrying cameras and bait lowered quietly onto the seafloor for periods lasting 1 or 2 days before automatic release and retrieval. These systems observe deep-sea animals without the continuous noise and light associated with active submersibles, sometimes capturing chimaeras moving naturally through undisturbed darkness. Yet, even after all these advances, the scale of what remains unseen is immense. Across every research institution, every expedition, every ROV survey, and every recorded observation, the total accumulated footage of wild chimaeras would occupy only a few hours of video. For a vertebrate lineage older than forests, dinosaurs, and mammals, the visual record remains astonishingly small. Fragments of movement gathered slowly from one of the least accessible environments on Earth, far below the surface turbulence of the world ocean, beneath storms, tides, coral reefs, shipping lanes, and sunlight itself, there exists an environment defined less by change than by continuity. At depths beyond 1,000 meters, the sea settles into extraordinary physical stability. Temperatures remain near freezing across immense spans of geological time. Salinity changes only slightly. Pressure remains constant, immense, and unrelenting. In this vast, cold darkness, evolution appears to move differently. The deep ocean functions as what biologists sometimes describe as an evolutionary refuge, a place where ancient body plans can persist for immense stretches of time because the surrounding environment changes so slowly. Near the surface, life exists inside a continual cycle of disturbance. Seasons alter productivity, storms reshape coastlines, temperature fluctuates across years, centuries, and ice ages. Predators evolve sharper vision, faster muscles, more efficient hunting strategies, and prey species respond in turn, creating endless evolutionary escalation. The deep ocean strips much of that away. Below the reach of sunlight, there are no visual horizons to dominate. There is little advantage in evolving brilliant coloration when darkness absorbs nearly all visible light. There is no annual bloom of food abundance followed by famine because food is sparse almost all the time, descending slowly from above in the form of marine snow. In such an environment, rapid metabolism becomes dangerous rather than advantageous. Energy efficiency matters more than speed. Stability matters more than innovation. The chimaera appears to embody this pattern more completely than almost any other vertebrate lineage still alive. Over hundreds of millions of years, its basic body structure has remained recognizable, while entire dynasties of marine animals rose and disappeared around it. Placoderms vanished, mosasaurs vanished, plesiosaurs vanished. Most ancient shark lineages vanished. The chimaera remained, drifting through deep water where evolutionary pressure itself may have softened into something slower and more conservative. It is not entirely alone in this refuge. The coelacanth, another lineage once believed extinct, inhabits deep volcanic slopes in the Indian Ocean and Western Pacific. Its fleshy lobed fins resemble structures known from fossils more than 300 million years old. The frilled shark, living along continental slopes at depths similar to many chimeras, still carries a body plan strikingly similar to Cretaceous ancestors preserved in stone. Again and again, the pattern repeats itself. Extreme depth becomes associated with evolutionary persistence. The irony inside this pattern is difficult to ignore. The deep ocean is among the harshest environments on Earth. Pressure rises high enough to crush unprotected machinery. Temperatures remain only a few degrees above freezing. Food is scarce, dispersed, and unpredictable. Absolute darkness stretches across distances larger than continents. Yet, over geological time, this hostile environment may have become one of the safest refuges life has ever known. Surface ecosystems experience catastrophe directly. Asteroid impacts darken skies and collapse photosynthesis. Climate shifts alter coastlines and weather systems rapidly. Shallow marine habitats disappear beneath changing sea levels. The deep ocean absorbs these shocks differently. Not untouched, never untouched, but buffered by volume, isolation, and thermal inertia. Catastrophes arriving from above descend slowly into the abyss, weakened by distance and time. This creates an unsettling possibility. If ancient vertebrate lineages have repeatedly retreated into deep water to escape extinction at the surface, then the modern deep ocean may still contain biological histories almost entirely hidden from science. Vast sections of the Indian Ocean floor remain unsurveyed. Thousands of seamounts have never been visited by remotely operated vehicles. Entire ridge systems stretch for hundreds of kilometers without a single biological survey. Somewhere within that darkness, there may be additional branches of vertebrate history still moving quietly through cold water, carrying genomes shaped in vanished oceans beneath vanished skies. The chimaera suggests this possibility not through speculation alone, but through its own existence. A fish, older than forests, older than dinosaurs, older than the first vertebrates to walk onto land, still survives because the deepest parts of Earth changed more slowly than the world above them. Somewhere in the ancient Devonian Ocean, long before forests spread across continents and long before vertebrates walked onto land, a small jawed animal moved through shallow water beneath a dim prehistoric sky. It possessed the anatomical foundations that would eventually give rise to sharks, rays, reptiles, birds, mammals, and human beings. The chimaera still carries traces of that distant ancestor inside its body, not as a symbolic connection or poetic comparison, but as literal biological continuity written into genes that have persisted across hundreds of millions of years. The chimaera is often described as a living fossil, yet that phrase can obscure something deeper and more intimate. This animal is not merely ancient, it is a surviving branch of the same evolutionary tree that ultimately produced the human nervous system, the human skeleton, the human immune system, and even the enamel covering human teeth. Beneath the unfamiliar appearance of the chimaera lies a body assembled from many of the same molecular instructions that shape every jawed vertebrate alive today. Genomic research has made this connection clearer than at any previous point in scientific history. By comparing the slowly evolving genome of the chimaera with the genomes of bony fish, amphibians, reptiles, birds, and mammals, scientists have been able to identify which genetic features were already present in the common ancestor shared by all jawed vertebrates approximately 420 million years ago. The chimaera functions almost like a biological reference point, preserving ancestral sequences that changed more rapidly in many other lineages. One of the clearest examples appears in the development of teeth. Human teeth and chimaera tooth plates seem radically different in form and function, yet the genes regulating their development belong to deeply related ancestral gene families. The molecular machinery guiding the mineralization and patterning of human teeth can be traced backward into systems already operating in ancient cartilaginous fish long before mammals existed. The structures themselves changed across evolutionary time, but parts of the developmental blueprint endured. The immune system reveals another layer of this shared history. Modern humans possess an extraordinarily complex adaptive immune system involving specialized T cells, antibodies, signaling proteins, and genetic recognition pathways refined over immense evolutionary timescales. The chimaera possesses a simpler version of this architecture. Certain immune components found in all bony vertebrates are absent, which means those features evolved after the chimaera lineage diverged from the ancestors of sharks and tetrapods. Because of this absence, researchers can reconstruct the sequence through which the vertebrate immune system gradually assembled itself across geological time. The skeleton carries similar clues. Human bone appears fundamentally different from the flexible cartilage skeleton of the chimaera. Yet the molecular processes involved in initiating mineralization show striking similarities. Some researchers now believe that bone may have evolved from earlier calcified cartilage systems rather than appearing as an entirely separate innovation. In this interpretation, the chimaera preserves an intermediate condition, not halfway in an evolutionary sense, but representative of a deeply ancient stage in vertebrate structural biology that later lineages modified into fully ossified skeletons. Even sensory systems reveal echoes of shared ancestry. The organization of vertebrate eyes, the developmental pathways guiding cranial nerves, the arrangement of jaws and paired appendages, all emerge from inherited anatomical frameworks established long before the first reptiles or mammals appeared. Evolution did not discard these foundations. It built upon them repeatedly, altering proportions, adding structures, and refining functions while preserving the underlying architecture. Seen this way, the chimaera becomes more than an isolated curiosity drifting through deep water. It becomes evidence that the human body itself is ancient beyond ordinary comprehension. Every heartbeat, every immune response, every tooth forming inside a child's jaw carries molecular histories extending backward into oceans inhabited by creatures that no longer exist. The connection is separated by 420 million years of independent evolution, by mass extinctions, continental drift, and the rise and fall of entire ecosystems. Yet, the relationship remains physically present inside living tissue and encoded within the genome. In the body of the chimera, fragments of the biological world that eventually produced humanity still persist, moving silently through darkness beneath the modern sea. Try to hold 400 million years in the mind, not as a statistic, not as a scientific label attached to rock layers and fossils, but as duration. A continuous chain of living moments stretching backward beyond ordinary comprehension. Every individual chimera that has ever drifted through deep water belonged to an unbroken lineage extending across spans of time so vast that human language struggles to contain them. Modern humans, in anatomical form recognizable today, have existed for roughly 300,000 years. The chimaera lineage is more than 1,300 times older than our species. Recorded human history, from the first cities and written records to the present day, occupies approximately 5,000 years. Against the timeline of the chimaera, the entirety of civilization becomes thinner than a surface scratch on stone. While this lineage persisted, the Earth itself transformed repeatedly. Continents wandered across the surface of the planet by thousands of kilometers. Ancient seas vanished, and new oceans opened between separating landmasses. Mountain ranges rose skyward and slowly eroded away grain by grain until little remained. The atmosphere shifted composition multiple times. Oxygen levels climbed, fell, and stabilized again. Forests of primitive ferns spread across swamp continents long before flowering plants existed. Giant insects crossed humid carboniferous air. Reptiles emerged, diversified, and dominated the land. Dinosaurs appeared, ruled for millions of years, and disappeared. Mammals inherited the altered world afterward. Through all of this, the deep ocean remained astonishingly stable. Below 1,000 meters, sunlight never penetrated. Temperatures remained near freezing. Pressure remained immense and constant. Food descended slowly from above in an endless drift of organic debris. In that darkness, the chimaera lineage endured, not untouched by extinction events or evolutionary change, but buffered from the violent instability that repeatedly transformed surface ecosystems. There is something unexpectedly calming in that continuity. On land, survival often appears tied to expansion, speed, competition, and continual adaptation. Species rise rapidly and disappear just as rapidly when conditions shift. Entire ecosystems rearrange themselves across relatively short geological intervals. The chimaera followed a different path. It did not conquer every ocean depth. It did not diversify into thousands of forms. It did not dominate the surface world. Instead, it persisted quietly within an environment that demanded patience, efficiency, and restraint. Everything human beings normally consider ancient collapses against this scale. The pyramids of Egypt, weathered and immense from a human perspective, belong only to the most recent fraction of the chimera's existence. Ancient Rome, the earliest dynasties of China, the first agricultural settlements along river valleys, all vanish almost instantly when placed beside a lineage already ancient before the first tree rooted itself on land. Even the evolutionary story of humanity occupies only the closing seconds of this timeline. Australopithecines walking upright across African grasslands, early Homo species shaping stone tools, modern humans painting cave walls and crossing continents, every step unfolded while the chimaera continued moving through cold water beneath the surface world, following behavioral patterns shaped in oceans unimaginably older. The deep sea changes the meaning of survival itself. On the surface, success is often measured through visibility, abundance, territorial expansion, or dominance over competitors. The chimaera suggests another possibility entirely. Persistence may depend less on conquering environments than on fitting them so completely that large change becomes unnecessary. 400 million years of survival emerged not from aggression or complexity alone, but from compatibility with a stable world hidden beneath the reach of sunlight. That idea carries an unusual emotional weight because it stands so far outside ordinary human experience. Human civilization moves quickly, driven by acceleration, growth, and continual reinvention. The chimaera lineage moved in almost the opposite direction, conserving ancient solutions across eras when entire biological dynasties vanished. In the darkness below the continental slopes, beneath storms, wars, cities, extinctions, and all recorded memory, the chimaera continues its slow passage through cold water. A living vertebrate lineage older than forests still survives not because the world remained unchanged, but because somewhere deep beneath the changing surface, one part of the world almost did. Beneath the familiar geography of continents and coastlines lies another planet-sized landscape that human beings still barely know. The deep ocean covers roughly 65% of Earth's surface, descending to an average depth of 3,688 meters. Yet most of that immense terrain remains unseen in any meaningful sense. Less than 1/4 of the seafloor has been mapped at high resolution, and only a tiny fraction has ever been directly observed by cameras, submersibles, or remotely operated vehicles. There is a strange imbalance in this. Human beings have produced detailed maps of Mars, tracing dried river valleys and impact craters across a world millions of kilometers away, while enormous portions of Earth's own seabed remain blank in scientific detail. Entire underwater mountain chains extend through darkness without visual survey. Vast abyssal plains, larger than nations, remain biologically undocumented. In many places, the ocean floor is still represented on maps by little more than broad estimates stitched together from sparse sonar passes. The chimaera exists inside this absence of knowledge. Its story is not only the story of an ancient vertebrate lineage surviving across geological time, but also the story of how limited human observation has been for most of scientific history. Until the 20th century, the chimaera was almost entirely invisible to science as a living animal. Even now, after decades of technological progress, most species are known from only a handful of specimens collected accidentally in deep trawl nets. Some species have never been filmed alive. Others are represented in museum collections by single individuals preserved decades ago. This creates an unsettling possibility regarding how much vertebrate diversity may still remain undiscovered in the deep ocean. Roughly 50 living chimaera species have been formally identified, yet the majority were discovered not through targeted exploration, but through incidental bycatch from fisheries, searching for completely different animals. Systematic biological surveys across most of the ocean floor barely exist. Large regions of the southern Pacific, deep Indian Ocean basins, and countless seamount systems remain almost entirely unsampled. Whenever researchers have looked carefully into deep water, the discoveries have repeatedly altered assumptions about what the ocean contains. At depths near 1,000 meters, scientists have found cold water coral forests stretching across continental slopes, ecosystems thriving in darkness far below the sunlit reefs once thought necessary for coral growth. Hydrothermal vent systems revealed entire biological communities powered not by sunlight, but by chemosynthesis, organisms deriving energy from chemical reactions around superheated, mineral-rich water emerging from the seafloor. Whale falls, the sinking carcasses of dead whales descending into abyssal depths, were discovered to support specialized ecosystems persisting for decades, feeding dense communities of scavengers, worms, and bacteria. Even seamounts, separated by only a few hundred kilometers, often contain dramatically different assemblages of life, isolated by currents, depth, and geography into distinct biological islands beneath the sea. Each new expedition seems less like filling small gaps in knowledge and more like opening doors into environments fundamentally different from previous expectations. Yet, the chimaera inhabits some of the least examined regions of all. Mid-depth soft sediment plains rarely attract the same scientific attention as coral reefs, hydrothermal vents, or volcanic ridges. To human observers, they appear visually empty, broad expanses of fine sediment descending into darkness without dramatic geological features. Research funding and exploration effort naturally drift toward more visually spectacular environments. The ghost shark remained within those quieter regions for hundreds of millions of years, moving slowly above seafloor landscapes that human beings overlooked almost completely. While surface civilization expanded across continents, charted skies, crossed oceans, and eventually reached space, the chimaera continued, inhabiting a world still largely absent from human awareness. There is something humbling in recognizing how recently this hidden geography entered scientific perception at all. For 400 million years, the chimaera existed beneath storms, empires, extinctions, and entire chapters of evolutionary history without observation. The animal did not suddenly emerge from darkness because it changed. The change occurred in us, in the gradual development of tools, finally capable of reaching the world where it had always been waiting. Far below weather, below tides, below the last fading traces of sunlight, cold water continues moving slowly through the dark, exactly as it did long before human beings existed. Somewhere tonight, along an unnamed continental slope, or drifting above a trench no person has ever seen directly, a chimaera is moving through that darkness, with the same measured rhythm carried by its lineage for hundreds of millions of years. Its broad pectoral fins rise and fall with soft precision, lifting it forward through water dense with pressure and silence. The long tail trails behind like a loose thread in the current, barely needed for propulsion. Around it, there is almost nothing visible at all, only suspended particles descending through black water toward the seafloor far below. Its eyes remain open, large and green beneath the dim glow of its reflective retina, though there is little light to gather. No moonlight reaches this depth. No dawn ever arrives here. The tapetum lucidum behind each retina waits for the faintest pulse of bioluminescence, the brief electric blue shimmer of another organism moving somewhere in the distance. Across the surface of the snout, the pores of the rostral organ continue sampling the water ahead, reading weak electrical disturbances produced by buried animals hidden beneath layers of fine sediment. Beneath the chimaera, the seafloor stretches outward in pale drifts of accumulated marine snow, organic remains falling continuously from ecosystems thousands of meters above. Inside that slow-moving animal is a genome carrying fragments of one of the oldest surviving vertebrate lineages on Earth. Within its cells remain genetic patterns, tracing back toward the common ancestor of every jawed vertebrate alive today. The architecture of its skull, its mineralized tooth plates, its sensory systems, even the chemistry guiding the formation of its cartilage, all preserve echoes of biological experiments that began before the first forests spread across land. When its ancestors were already navigating deep oceans, the continents themselves still occupied unfamiliar positions, and terrestrial ecosystems were only beginning to emerge from coastal mud. The chimera does not know any of this history. It is not ancient in the way human beings imagine ancient things. It does not move through the ocean like a relic conscious of survival. It is simply living inside the same environmental conditions. Its lineage entered hundreds of millions of years ago, following sensory signals through darkness, conserving energy, feeding when food appears, reproducing slowly, persisting without urgency. The deep ocean never demanded speed or spectacle from it. Stability became enough. There is something quietly recalibrating in that realization. Human beings spend most of their lives near the surface, surrounded by rapid change, noise, heat, weather, and constant motion. Civilizations rise and disappear within spans of time that barely register against the age of the chimaera lineage. Languages emerge, empires expand, technologies transform the world above the waves, yet deep beneath all of it, the same cold conditions remain almost untouched. The chimera invites a different scale of thought, one measured not in generations or centuries but in geological continuity. And after spending time inside its world, the animal no longer feels entirely alien. The shape of its body becomes familiar. The green reflective eyes, the wing-like fins, the slow searching movement above the sediment, all begin to feel less like something monstrous and more like another expression of vertebrate life, shaped by a different environment and a vastly different time scale. Much about it remains unknown, perhaps unknowable for now, yet it no longer exists only as a strange silhouette glimpsed in deep water footage. Somewhere between 200 and 2,600 meters below the surface of an ocean you may never cross, a ghost shark is gliding through darkness exactly as its ancestors once did, while the earliest forests spread across the continents above. The deep water still surrounds it completely, cold and lightless, holding its secrets with the same silence it has always kept.